Okay, good morning, everybody. We are about to start this session. I don't want to take away time from the speakers, so I'll just make a few introductory remarks. Some of you may have been wondering what is the title of the symposium, Evolutionary Ecology in the Wild. And I've been getting some peculiar remarks from various people. Are this like wild ecologists behaving wild? What's going on? uh, it actually comes from the title of a book written many years ago by a very eminent evolutionary biologist, and the title of the book was Natural Selection in the Wild. Now again, you may ask me, why natural selection in the wild? And the reason for that was that most evolutionary biologists have studied experimental evolution with Drosophila or C. elegans or all the other model laboratory organisms. Yes? So here we have a group of 4 very talented people who will take you through their systems or with ideas about how evolutionary ecology is really makes sense when you're looking at natural patterns that are occurring in nature. So without any further ado, I'll invite Uma Ramakrishna, she's a fellow of the Academy and from NCBS. Uma, the floor is yours.
Okay.
This is the forward, this is the backward. Doesn't hold this one.
That's a pointer. Oh, okay. So good morning, everyone. I'm Uma, and I'm going to start. This is a new talk, so hopefully it'll be great to hear what you think about it. So as you all know from our high school textbooks, India and the Indian subcontinent has had a really interesting past. As an island, it made the longest journey of any landmass across the oceans until it hit Asia several millions of years ago. At the same time, recent data, this is a meta-analysis by Allen and others, shows us that basically if you look at the predicted risk of zoonoses or spillover of infections from animals to humans, India appears to be a hotspot, a potential place where a lot of zoonotic spillover, a lot of pandemics potentially like we had just recently, COVID-19, will happen. So I'm going to try and connect these two kind of facets of science. How do we actually study this? What are the consequences of the movement of the Indian plate on Indian biodiversity? And how does that then result in what we potentially see or what might happen in the future? This is basically a field of, biogeography, and I'll try and connect that to zoonoses towards the end, and hopefully you'll see a link. That's how I got to studying disease ecology and zoonoses that I do more of today. So I'm going to focus on small mammals. Mammals are our closest relatives, and half of all mammals are small mammals. In this case, I'm going to talk about rats and mice, old world rats and mice, and some shrews. So basically, biogeography is very simple, and it's a very old form of natural history where people like Darwin and many others, Wallace, Humboldt, why species are in specific places, which species are where and why. We can also think of this from a perspective of communities. Once a species is in a location, who else is around it? What species do they interact with and compete with potentially, right? Disease ecology is, you have all these species in a community, and then there are microbes laid on top of that, right? So for the microbes, the community of, say, rats and mice is their habitat. and they kind of move between species. For example, that's host switching or potentially spillover if the species in question is, say, a human, right? So basically, I'll take you through a few studies here and then talk in the end about how in the future we hope to look much more specifically at zoonoses. Okay, so just zooming out, when we talk about which species are where, let's take a context check. We are in India, that's over here, and I'm sure you hear in the paper all the time, India has high biodiversity, India has high biodiversity. Well, for mammals, not really so. Southeast Asia, in our neighborhood, has much higher biodiversity, but of course there are some locations, for example, the Northeast has really high biodiversity. That's the comet that you see kind of going into India is the Himalayas, right? So basically it seems like, India has reasonably high biodiversity, but of course its neighboring areas seem to have more. So can we deconstruct this pattern? Of course, it has something to do with India's past. The fact that when mammals were speciating globally, India was an island floating through the ocean, kind of isolated from all of these processes. So if we look at that from a more quantitative perspective, what we can think of is that maybe India is simply a sink with species after it hit Asia coming in from the west, the north, and the east. And so we tested this with patterns, just patterns. This is called a macroecological approach. We just say grid up the whole of India, look at species in these grids, and plot these grids as a function of, where are most of the species, say, in a genus from. And it's really beautiful. It's like India is a blotting paper, and you have ink coming in from each side. And as you get farther from the sources, that ink or the number of species dilutes. So you can see here, for example, this is Western taxa, and you can see it's highest in the west. This is Eastern taxa, highest in the east. Note that the numbers are much higher. Sorry, it's getting cut off, but this is like much higher. The red is much higher here. And this is Northern taxa. The Himalaya, like a step function, they just don't get beyond the Himalaya at all. So does this mean that there's no, there's very little kind of endemic diversity in India? Is everything in India we have somewhere from outside? So how do we test this? Well, what we've done is we've actually put together a phylogeny. This took several years, actually a decade or so, and we did it for a speciose group. When you want to look at a process, you want to look at it with many species, and we did it with these guys. They're murines, so basically old world rats and mice. And there's about 704 of them, 704 murines in the world, and we built a phylogeny with 437 murines, so 437 of these. And this is the largest phylogeny so far. This is not published yet, but basically the last phylogeny had only about 268. Of these, 40 species are in India, and we have sampled 34 of those. So over time, the lab has been working in various habitats, and we've been able to sample a significant amount of murine diversity. And what does such a phylogeny tell us? We can actually build this phylogeny and then basically ask, so that's the phylogeny. It's really messy because obviously it has so many taxa, right? It's like very, very long. You can't see much, but hopefully you can see these colored like nodes, and the colored nodes are what is called ancestral reconstruction of these areas. So the question we ask is, the tips, which are the Indian species, where are they from, right? And what we find is, as expected, so basically this grayish blue is Southeast Asia, the big kind of dark blue is the Palearctic, and so on. And you can see basically that kind of as expected, sorry, this seems to have moved a bit. Most species are Southeast Asian, about 60% or so of Indian species are from Southeast Asia, right? So in terms of origins, species have come in from into India. There has not been a lot of diversification or speciation within India, and that is that also the phylogeny shows us. In fact, there have been more back radiations, species going back from India into Southeast Asia than there have been endemic species radiations. There's many questions that remain. Basically, did this happen at some specific points in time when the climate was conducive, wetter, so on and so forth? But this is something which we're still investigating. Okay, so that's fine. But I told you that I'm talking about communities, right? So this is some work which we completed recently, and it's looking at communities and disease in the context of the Western Ghats. I say right off the bat, this is potential pathogens. We have not tested pathogenicity of these microbes at all. What we did is, we now know, since we now know, that most species are potentially from Southeast Asia in a community, we looked at a biodiversity hotspot, the Western Ghats. We sampled 2 sites, Kadumane, which is a recovering plantation, tea plantation, and Kudremukh, which is a protected area for small mammals. How do we do that? We basically have traps, these little traps, where we bait them with butter, so the small mammals are attracted to them for food. But the traps could be up in the trees, as you can see here, or on the ground, and we set them up in a grid, and basically we catch animals, and you can see how many ectoparasites this animal has. Can you see all the blood spots on its ear? So these animals are living with many, many ectoparasites, which we basically sample both the ectoparasites and the small mammals to look at what microbes they co-occur with, right? So we basically can do some measurements, tissue samples, blood samples, oral swabs, rectal swabs, and look at endoparasites. And basically the methods are pretty simple, standard. We get these samples, both ectoparasites, sometimes if animals die, sacrificial, we take spleen, and then we basically look at, we extract DNA, we do PCR. In this case, we looked at a potential pathogen called Bartonella. It's a mild potential pathogen, unlike viruses, which are very dramatic. It causes something called cat scratch fever. You get these rashes, and it's actually very common in forest-associated communities, but it's never diagnosed. So this is an example of a potential spillover. Oh, it started vibrating for some reason. And basically, something which is usually probably undetected, right? Another very good example of this is Q fever or scrub typhus. Again, very often found in forest-associated communities, almost never diagnosed because usually it goes away, it's not really serious, a fever for a few days, right? So basically, in terms of the rodent community, that is, we have various habitats here. Like I mentioned, we have grassland, we have forest, we have built-up area, the labor lines where people are working. And what we see is that this is just showing you which species, these are just species of small mammals. I mean, these are rats, for example, these are mice, etc. You can see that, for example, when you look at community structuring, grassland communities are different. So the grassland is GL, are different from forest communities, are different from built-up communities. Okay, so basically, not just are there different species of rodents, say mice prefer to live in grasslands. This is a better way to visualize it. Basically, there are forests, there are grasslands, and there's built-up areas. Rattus rattus is our commensal black rat, right? And we have Rattus satare. It's an endemic rat of the Western Ghats. But you can see there are what we call shuttle species, these species which co-occur in different habitats. And these guys and their ectoparasites will be the one which can potentially cause spillover or movement of microbes in that community and landscape, right? So now the question is, how do we test this? So far, it's ecological data showing that there is this community. Some species are more common in certain locations. But what we can then do is, of course, just to show you some nice pictures, these are ectoparasites. And we know very little about, they're not all insects actually, some of them are not. But basically mites, ticks, fleas, we can't even classify them, but we do sample them. And we are assuming they're the vectors. So we look at the Bartonella from the ectoparasites, from the animals, and build a phylogenetic tree to see, are specific ectoparasites associated with specific species, and that is reflected through the Bartonella or not. So first of all, in terms of prevalence, overall the prevalence is really, really high. So in the community, that is 60% of the small mammals here have Bartonella, right? And in fact, some of them have really high prevalence. For example, Rattus satare and this CF fernandoni, we'll get to that in a bit. Don't have to focus on the names. But when we look at Bartonella prevalence overall, it is dependent on ectoparasite load. So that suggests that it is these ectoparasites that are transmitting the Bartonella. Now, Whether this is pathogenic or not, we'll get to that in a minute. But if we also try to, again, reconstruct the origins of the Bartonella associated with these different species, you see, for example, overall, yes, you see specific Bartonella only occurring in, say, Rattus satare, but you also see an example here of a host switch, where a Bartonella from Rattus satare has moved to this Fernandoni mouse, okay? And remember, the Fernandoni mouse was part of the switch, part of the species which could shuttle or, transmit things because it was in multiple habitats. So here's a phylogeny just to show you the lineages in brown are known pathogenic Bartonella, known to be pathogenic to humans. We don't know anything about pathogenicity in animals. And you can see that many of our Bartonella strains are very close to pathogenic strains. So there is potential that the Bartonella we have sampled in these small mammals are pathogenic. We have not tested it. That would require culturing the bacteria, infecting cells, and so on, which we have not done. And also, basically the ectoparasites carry the same Bartonella strain as the animal they were on. So that shows that it's basically is being transmitted by these ectoparasites, and it's not some strange, funky thing that's going on. Okay, so to summarize, basically Indian mammals are primarily Southeast Asian in origin, and we tested this with both macroecological and biogeographic and phylogenetic analyses. And mammal communities host many potential pathogens, sometimes with very high prevalence. For example, though, when we look at scrub typhus, which is caused by another bacteria, it's not as high in prevalence. And so these disease ecology studies can be quite difficult because you require many, many, many samples to actually get at levels of prevalence. So that's why, again, it's vibrating, but that's why a lot of people focus on something like Bartonella. And for example, Robin's lab works on avian malaria because the prevalence is quite high. And it's easier to do science when the prevalence is high. If, for example, for a species, for a potential pathogen prevalence is really low, it becomes really difficult. Okay. So now I'll just touch a little bit on some work we are doing, and clearly I'm going to finish very early. I'm sorry about that. But basically, some time ago, I worked on a very student-led idea of a bat harvest. So in a place called Mimi, which is very close to the border in Nagaland, close to the border with Myanmar, there's a community called the Bomar, and every year they harvest bats from specific caves. So basically they smoke caves, and then the bats fly out, and they kill the bats, and then they are exchanged as gifts as a tradition, and this happens year after year. So we thought this would be an interesting interface to look at this potential of spillover, because it's a high contact interface, right? There's a very high contact between humans and when people get bitten by these bats, and so on and so forth. And as we know, bats carry a lot of potential pathogens. They have really amazing immune systems. They're immunosuppressed, and this is suggested as one of the reasons why they live so long, because they don't have immune reactions to pathogens, especially in intracellular pathogens. They're not so good at dealing with extracellular pathogens. So basically, we sampled these bats and these humans, and we actually looked at serology to look at whether there's shared serological response between bats and humans. And we found shared serological response for several filoviruses. So filoviruses are, the famous filovirus is Ebola, right? But this is very surprising because there's no Ebola, there's no filovirus actually known from Asia at that point. They're only known from Africa. And so this study caused quite some commotion, and there were several other things which happened. But overall, it also helped me realize with a jolt that, while we're talking in the Western Ghats about 10 species or something like that, in the Northeast, there are many, many more species. If you remember that figure of, the diversity, first of all, 60% of our diversity is from, Southeast Asia, which is the gateway for that is the Northeast, right? And also, basically, there's a lot more species there. So if you remember the plot, and if you, basically 2/3 of India's mammals are in the Northeast, okay? So most of India's diversity is actually tilted towards the Northeast. So what we've done recently is we've actually started like a large program, and we just started, to actually try to look at not just wildlife, but also livestock and humans. So a sampling site in this case is, caves with bats, forests with bats associated with the caves, villages associated with those forests and those caves, and their agriculture and jhum. They have shifting culture, shifting burn and slash and burn and shifting agriculture. So those sites, and the idea is to basically sample, this is just like, there's lots of institutions involved, but basically to sample these species for associated viruses and bacteria, and to sample humans as well for the same. So the idea is then we can actually look at not just what are all the viruses and bacteria in the bats, in the wildlife, in the livestock, but also in humans. And hopefully just to show you, these are some of the habitats, extremely difficult field work. A lot of the caves are at the base of these cliffs, and a lot of this is Meghalaya. And basically, like I said, we just started field work, so it's really fun. We have, this is Vivek, who's done a lot of work on small mammals. This is one of our person in the field team. She's holding a bat here, you can see. And you have to be very careful with these gloves and wear masks because, like I said, bats have many potential pathogens, right? And this is a small mammal called a niviventer. And we basically sample them, like holding like this for oral swabs, rectal swabs, wing punches, and so on. And the idea is hopefully to come up with some kind of a framework like this. So this is a very nice framework published a few years ago, which talks about risk. So you saw from the perspective of that community ecology, disease ecology study, it's very ecological, right? It says, okay, there's Bartonella in the system, there are these species, there are some species which have higher prevalence of Bartonella, some of those Bartonella are potentially pathogenic. So if, for example, we went to community health sites close by, like PHCs, and sampled people, maybe we could look at which of those are potentially pathogenic or spilling over to, say, the people who are working in the tea plantation, for example, right? But it's still very small scale. It doesn't really tell us much about, say, the risk of spillover more generally. So the idea here is to understand what's called hazard, and hazard is basically what bats are there, what rodents are there, what bacteria and viruses do they carry, how potentially pathogenic they are. So for example, I'm almost done. Basically, for example, coronavirus is not very pathogenic, whereas Ebola is very pathogenic, right? So like that, basically, what is their potential pathogenicity? But what's equally important is exposure. How are humans interacting with wildlife? When are they interacting with wildlife, right? And that we are trying to assess through surveys, and also looking at basically risk, which is the interface of these two through serology. So we are working together with Rahul Roy at IAC. He's developing some new multiplex serology, which can be done with really tiny amounts. In fact, we are actually collecting blood spots, so not even sera, which is what is usually used for serology. You have to have sera. And basically, we're trying to understand risk and how this risk varies across space. So the goal is to sample about 180 sites across Arunachal, Nagaland, and Meghalaya, and then to basically look at risk, calculate risk, and see, are there some places where there's more risk and others where there's lower risk, and then maybe design some kind of a way forward to think of how do we monitor this risk. Do we have to go and study every single bat, find out every single detail, or can we make some generalizations and educated guesses? So in general, basically we have some understanding communities is critical for biodiversity, disease ecology, and emerging infections. So the way we've always treated zoonoses is post hoc. There's an outbreak and then we rush to figure out who is patient 0, where did this spill over from, how did it happen? But it would be really nice if we could understand beforehand where are areas where there's a high risk of spillover, for example, right? And so this is what we're hoping to do. And as I said, I hope that came across, but while there's a lot of studies in the Western Ghats in general in ecology, a lot of times we are missing, for example, the Northeast, which is really where all the action is. There's a lot of biodiversity there. And so we should be working in such landscapes, and also a lot of human-wildlife interactions, right? And so basically, fortunately, unfortunately, this cannot be done by AI. We have to do on-the-ground, field work, collect some. Of course, analysis, AI can aid in analysis. There's going to be humongous data sets, but it will require boots on the ground sampling and field work, something which is quite often disappearing from a lot of research now. There's a lot of data and people are just analyzing data. And so, that's probably a philosophical point, but I think this is also really important for the future. I have to thank Partha, who helped us in kind of securing this funding through DBT. So the work I presented was that of Ansil. He's really a fantastic, he was my PhD student. He's now doing a HFSP postdoc looking at bat coronavirus and immunity in Costa Rica. And this is Vivek. I presented some of his work as well. He was also a postdoc with me and now works with the MSc Wildlife Program. The phylogenetic analysis, the big murine phylogeny, was put together by Chetna, an undergraduate at IISc, and Aparna Lajmi, who was a PhD student at IISc but also worked with me briefly. Thank you so much for listening, and happy to answer questions.
Thank you. I'm sure there are lots of questions. Yes?
My clarification is in particular with your first part of the talk on biodiversity, where you mentioned that many of these species move from Southeast Asia or northern part or the western part. Over the years, when you make this phylogenetic tree, did you found that there was cross-matting in these species, and you found some new species which are being evolving while they are within the country?
So that's a very interesting point. Actually, that would recently, in general, there has been many discoveries of hybrid speciation, where two species hybridize and create a new species. Generally not at all common in mammals, much more common in plants, for example. We have not found any evidence of that. It really seems oddly that India is kind of empty of there's not that many endemic species in India. So maybe that was because there were a lot of extinctions. We don't know what that is because of. But one of the things is that, Southeast Asia is extremely wet. So there's a lot of historical contingency. That is, species that have evolved in wet environments tend to stay in wet environments, right? Whereas species which are from the west are more arid, dry zone species, they tend to stay in more drier environments, and so on. The Himalayan guys never get past the Himalayas, right? They're used to more cold environments. So in India, there's a lot of gradients driven by lack of rainfall, and that kind of prevents probably some species even from meeting each other, as you would say. But communities are dominated by Southeast Asian. Communities and numbers are dominated by Southeast Asian species.
My question is that when India broke off from the Africans continent, is it that there was nothing, no
There
mammals,
were some species, and
so
those
what
are
was that fraction
Right.
that still persists to this day?
not mammals. So there were, because mammals evolved
Much later.
after that,
Okay.
while it was floating. But the classic example is the purple frog, Nasica batrachus, which is basically a Gondwanan species when India was part of that larger landmass. But for mammals and birds, most of the speciation happened after KT, 65 million years ago. They were around, but they started speciating after that. And so India kind of missed out. There was also a lot of volcanic activity on India as it was floating. So, yeah.
So this is just a continuation actually of his question. So
Yeah.
are there species that have evolved in peninsular India which we can say are just...
There are some, but
Yeah,
very
but
few.
very few,
Very
you're saying.
few, very
Okay.
few.
And this
So-
is true for across, I mean,
No,
mammals
for
and other-
mammals, birds, for example, Robin has found some endemic radiations, but generally very, very few.
We have time for Vinod.
The Himalayas have risen over the last
Yes,
50
yes.
million years.
Yes,
And when they touched Tibet,
Yeah.
there was some migration from the north,
yes, yes.
particularly of mammals.
yes.
And as it began rising,
Yeah.
there might be an evolutionary sequence of processes
Definitely.
That
yes.
distinguish them, between what exists in the foothills and.
Right. So from what we have studied, I didn't present it, we have done some biogeographic work in the Himalayas that suggests more patterns of dispersal. So basically the Himalaya is like this. The eastern Himalaya is more tropical and the western Himalaya is more temperate. So we see patterns of dispersal and a little bit of speciation within, but not intense speciation in the Himalayas. So it's more a pattern of dispersal, but we're going to be sampling Arunachal, Nagaland, Meghalaya. Hopefully we'll get more. We need to do more sampling. We really don't even know the species of rodents in the Himalaya, very frankly. It sounds kind of silly, but that's how it is.
Here. Okay,
yeah, just a quick follow-up question on the geoecology.
Yeah.
Madagascar and Western Ghats.
There are some shared species.
The flora fauna must be, the biodiversity must
No.
be. No?
No.
There's no.
Of trees, yeah.
Yeah, it's been studied.
Yes.
Fully.
That's in
Okay.
fact one of the proofs of plate tectonics
Right.
is, the fact that there are shared species across these very currently disparate continents. But definitely
this
mammals.
was the breakaway of Palakkad Gap
Huh.
that created Madagascar. So
No. No.
that's the popular version.
No.
That the Palakkad Gap was actually created by the breakaway
No.
of Madagascar.
No.
No?
I don't think so.
That's
here.
Yeah. Hi.
If the mammalian evolution more or less happened after India broke off from Africa,
Yeah.
then
Globally.
how come you have similar species like hyena, like rhino, like elephants?
Those are all dispersed, yes. So for example, rhinos are very, very widely distributed, right? And they are larger. So we're talking about small mammals. Their ability to disperse is much lower. So their distributions in general are smaller. But these species, you're right, hyenas, crazy wide distribution, right? Hyenas, wolves, leopards, these are all larger species and they have potentially dispersed. But definitely there are still patterns, like tigers are Asian, they've evolved in Asia, most probably South China, whereas lions have evolved in Africa and have dispersed. So phylogeography that is looking at patterns of genetic variation in populations across space can allow you to infer those patterns of dispersal, the directionality and timing.
We have 12 seconds for one very quick question.
Raghav.
So many of the Nipah outbreaks are on the
Yes.
western coast, not in the northeast.
Yes.
say, some
Yeah.
reason.
So basically, the Western Ghats, so that map, I just showed it, the determinants of high probability of zoonoses is high mammal biodiversity, high livestock density, high human density. And the Western Ghats is a biodiversity hotspot with the highest human population density in the world. So yes, definitely there is zoonoses in the Western Ghats as well. But I think, I like being an explorer a little bit, and the Northeast is really very, very unknown. A lot of data from China, and so that's nice to compare, actually. A lot of similar species. So hopefully we'll find some interesting things.
Well, sorry?
There's a student at the back.
A student, yes.
Hi.
Hi.
Yeah. So very nice talk. I just had a very quick question. So the local populations which are in close proximity to the wildlife there,
Yes.
over the years or maybe over the last, how many years they are together, they might have grown immune to some of these pathogens. Do you think some of these things might be different for the mainland or maybe other parts of the world? And how are you going, is it something people can potentially study to see that maybe the locals are not something who might really be
Local
affected
people, you
badly?
mean? Local
Yes,
people.
yes.
Yeah, so basically that is probably very true. There's a lot of signatures of in the genome of humans of adaptation to, for example, malaria and so on. Unfortunately, in this project, we didn't get the funding and permissions to look at that for humans. Maybe it's something we can do later. There's a lot of biology also, for example, understanding the pathogens and so on. But yes, that's a really important point. Yeah. Thank you.
Okay.
Thank you so much.
Thank you, Uma. So now we come to the Western Ghats, yes, with Robin, who will tell us about birds. Robin is from IISER Tirupati, and we hope that he will do a lot of work in the Eastern Ghats now.
Person hold this one, digital point will appear on the screen.
Hello everyone. Thanks for inviting me here, and I'm happy to have you all here at IISER Tirupati and to be talking to you all about some of the work that we do. I'm going to just bring together a few sets of data that that I'm very, very excited about. This is probably just the second time that I'm trying to put this together, so I'm happy to hear from you if there are any gaps of logic that you see. So this habitat that you see here, there are a few things that I'd like you to take away from this. The first is that this is the Western Ghats, the highest elevations of Western Ghats. That's the Anemudi peak that you see there. The other striking thing about this image is that you can see two kinds of habitats here. One is the grassland, and then you have the forest. And the important part is that both are natural. So people have done, they've dug cores. In fact, Professor Sukumar from IISc was one of the people who led that idea. And they find that grasslands are natural here. It's not because of deforestation. So there are some parts of the landscape that are occupied by forests and some that have grasslands. So this is an important part, and it will kind of become clear as we go further ahead. I want to first, right up front, acknowledge that a lot of this work was led by Meghna Natesh and another student, Vinay. And then a bunch of PhD students came along and contributed to various other parts. And finally, Rais, a BSMS student, has kind of put in a few important parts to the story that I'm going to present. So my group primarily works along four themes. First is, first is this part about the vegetation or habitats, and then bird evolution and biogeography, some of the kinds that Uma was also talking about. But I also focus on bioacoustics, bird song, and how songs change and behavior. And finally, citizen science and outreach is also something that we've been kind of dabbling with a little bit. So the first thing that I want to kind of explain here is what is a sky island. I think everybody knows about the idea of what an island is, because it's area, any kind of landmass that is isolated by ocean usually. But in case of sky islands, these are mountain tops, and they are actually isolated by valleys where the climate is different. So it's the climate, not the oceans, that separate one habitat from another. So one mountain top from another is separated by the valleys, which have a very different kind of climate. Okay, and how important is this? The proof of that is in the Western Ghats. You have these, the mountain tops are in the dark color in the map, and you have different species on different mountains, and each are about 1 million or so years apart. And this one here is the Palghat Gap that we were talking about in the questions before, and that has led to a differentiation about 5 million years across the in these species, okay, across the Palghat Gap. So just to kind of tell you that the forest birds in these landscapes are isolated to these mountain tops, and I spent my first 2 years of my career actually walking up and down these mountains. So that's from 2000 to 2002, and 550 kilometers or so of transects. And you don't find these birds below 1,200, 1,300 meters. So at that time, we didn't have genetics, and that's when I kind of met Uma, and then we kind of we got the story together that there is this isolation for millions of years across these mountains. So these are like islands. So these mountains are like islands where there are many species. So that's the starting point of that idea. And climates are what climate is what separates these islands apart. What's also interesting is that grasslands are also, they have their own unique species. For example, this species, Nilgiri pipit, is about 7 million years old, which means that that part of the habitat was also occupied by a unique kind of fauna in this landscape. So you have forest birds, grassland birds, endemic, found only in these mountains, separated by climate. So coming to climate itself, what these two figures, these maps show, I think if I double click that, it gets out of slideshow. Yeah, is that this time called last glacial maxima. I think everybody's heard of Ice Age, especially movies and things like that. So that's when there was a lot of ice formation in especially in the polar areas. What very few of us know is that in the tropical parts, ice never reached us, but it became very dry. Okay, so we didn't have ice, but the habitat generally became drier at this period, and this was the maximum aridity that was seen in the recent past. Okay, that is the 20,000 years back. So the question then is that we have this landscape which has forests and grasslands, and we have this constant fluctuation of climate, especially in the last 2 million years or so. And this data comes from cores, sediment cores, that people have shown this globally, that climate has fluctuated quite drastically over time. And what about India? We know that globally grasslands have kind of expanded at the LGM, which is the LGM is what is this Ice Age, okay? Yeah, that's India there. And what you can see is that most of that habitat is, most of India was grasslands at Ice Age. And we again know this with soil cores from all of these areas. I just want to make the point that at LGM, it is generally known that there are a lot of grasslands in India, in the Indian landscape. And at this point, I want to introduce another idea to you. So in ecology, actually, there are very few rules, because there's always an exception, because there's so much heterogeneity and so many factors that come into play. But one interesting rule is this idea of what makes habitats. And there are two very interesting variables. This is called the Whittaker plot. And what it basically says is that temperature here on the x-axis and precipitation, the rainfall, a combination of this can tell you what kind of habitat is found anywhere on Earth. Okay, so you can predict, let's say, evergreen forests or any other savanna or other habitat very easily by just looking at temperature and rainfall. So essentially, one climate implies that you have one habitat. But then you have this exception, which is this Shola habitat that we looked at first, which is that you have one climate, but you have two habitats. You have forests and grasslands. So here's an interesting paradox, you can say, to this rule. And what I'm interested in is in trying to understand how birds have kind of coped with this two habitats, and how have things changed over the past climatic changes. And so here are all the actors in some sense. We have the forest birds and the grassland birds. They are and we now know that forest grassland mosaic might have changed over time. And this is a rough model that I started kind of putting together in 2011, and with a lot of discussions with Uma at that time. Idea is that as climate changes, as you go from wet to as you go from wet to arid to wet again, wet to dry. So the highest point of aridity is the LGM or the Ice Age, And that's when you think the population size, let's say, of grassland birds should be the highest, because we know that grass should have dominated the landscape. But then, because you have this forest grassland mosaic, then you would think that the forest should have declined at that point, because if the grasslands have expanded, grasslands have contracted. And you would find this kind of, it's almost like the forests are expanding, grasslands are contracting, and the grasslands are expanding and the forests are contracting. At least this is the model that we had in mind. And of course, there's one more component to this, which is that depending on the ecology of the species, depending on how specialized you are to the habitat, your amplitude of change would kind of vary. Okay? This is a little bit complicated idea, but I'll get to it in a little bit. It was in 2012 that there was this really cool genomic tool that came up called PSMC. And what it does is that from a single, this was primarily described from human genetic studies, where based on the coalescent theory, the idea, I collaborated with Nagarjun Vijay for this. And essentially, you can determine population size changes just from a single diploid genome. And primarily, it's coming from information of heterozygosity across the genome. So in very simple terms, I won't go through a lot of the details, less heterozygous, lower the NE or the effective population size. And more heterozygous it is, the higher the effective population size is. And you can get an idea of the change in effective population size. And this is the way that this data is represented. That's the past, and this is the current time. So it goes from the past to the current, and if population size is here on the y-axis. So you can see populations changed. There's an increase and there's a decrease and so on. So that's what we are going to be looking at. The first project that we did was kind of a proof of concept. This was led by Meghna again, and this really showed us that at the Ice Age, one of the species, the open habitat species, this was across 2 owls, one species increased exactly at around LGM, while another one showed a slightly different trajectory. So we thought that, okay, so now we have a method where we can actually kind of hit the Shola Sky Island species with this method. And we wanted to kind of sample as much of the community as possible. So this is the bird community of the Shola habitat. Many of these species, especially the ones with the orange stars, they are endemic to these mountain tops. So they are not found anywhere else, mostly on these mountains. But there are many other species like, let's say this, the bulbul that you would find widespread. They are found in large parts of the landscape, but they're also found on these mountain tops. Okay, so this is the community that we are looking at, once again, to kind of simplify the idea, we want to compare forests and grasslands, but we also want to compare the specialists and generalists. Okay, the species that are highly adapted to these mountain tops, and some that are not, some that are found widespread, found everywhere, and we would think that their responses might be different, and that's where this factor comes in, where the responses are slightly different. So we kind of assembled 45 genomes at various depths, essentially we are looking at effective population size over time. Yeah, I won't go into the details of that. But there's one thing that I do want to just briefly mention, and this is this cool idea of trying to establish how much of a specialist is a bird and how much of it is of a generalist it is. Okay, how specialized is a specialist and how much of a generalist is a generalist. And we did that by running these species distribution models, essentially plotting things like, let's say, the same temperature and precipitation axis, but you can see how much of the area does a species cover. So the more climatically specialized the species is, the smaller that area would be that a species covers, right? And if it is found across, let's say, large parts of the landscape in India, then it would cover a variety of climates, and hence that, that volume, that niche volume would be much larger. So that's what we calculated for every species that we studied. So this is just a graphic illustrating the same thing. You have restricted species on the mountaintop. They should have very small niche volume, and species that are found as you go from the mountain and you go further and further down, you should see that the niche volume that they occupies is larger and larger. So they are more generalists, okay? So that's the overall idea. And what did we find? Remember, we are looking at generalist, specialists, and forest and grasslands. I'll walk you through this. Essentially, what we find is that, so first the left corner, the grassland birds, the generalist grassland birds seem to show the pattern that we had thought would be found, which is that there is a big expansion and decline and so on. But the forest species, especially the specialists, showed no change at all. They were kind of consistent all through time. And the strange part was that the grassland specialists also showed no change. Okay, so this was really surprising to us. But strangely again, the forest birds, which were widespread, they did seem to show some change in population sizes. You have to remember that most of these end at about 10,000 years. So it doesn't tell you much about the recent past. It's all from about 1.5 million to now. So essentially, some ideas are supported, but some are not. But the cool thing that I think that we did was that Meghna calculated this fold change, let's say, trying to understand what is the amplitude of change, which is the minimum to maximum, how much is the change actually there in this population size. And what she found was that as the niche volume increased, which means that as you become more of a generalist and you occupy larger climates, your amplitude of change, the demographic change is more, okay? So essentially, if you're a climate specialist and you're on mountaintop, you don't seem to be changing too much. You're in a climatically stable place, yeah, and your population size is also very stable through millions of years, and that's what this shows. And that is irrespective of whether you're a forest bird or a grassland bird, okay? So that's quite interesting. I just want to say that we don't have very clearly discernible trends with timelines, that it doesn't completely match the LGM and so on, but that's a problem that we are trying to deal with. However, we simulated the data, which is that you can say that the population, what if the population changed from such to such, and then you create the genome and then push it into this PSMC analysis, and we are able to recover the same patterns. So it's not an artifact, but there may be a few other things that we need to do to figure out what exactly is happening. So the main thing seems to be that we have some partial support of our big model, and it is that ecology of the species seems to kind of determine the response to historic climate change. Yeah, and mountaintop specialists are stable over time. So remember, I started with this idea that we have one climate but two habitats, but what we seem to be recovering is that even if you have one climate and two habitats, one climate is resulting only in one response. So birds are responding to the climate, not so much to the habitat. And that's really interesting. And my personal, like what I really think is happening is that it's driven by clouds, because mountaintops have, especially the tropical mountaintops, get most of their rainfall from clouds. And clouds are known to be stable through time. So it's the stability of clouds that seems to be, yeah, okay, yeah. So very quickly, the second part of work that I wanted to talk about was how this landscape has changed. So this is just an illustration that shows that you had the forests and the grasslands, and then during colonial times, we went and planted some timber trees in it. This is like acacia and eucalyptus and so on. And this went wild because they are invasive species, and they started to cover up to 70% of a landscape just over recent time, just in a few 100 years. And although that was an illustration, I just want to say that we have a lot of satellite imagery and other data to where we have quantified this change that has happened. So over about 6 to 7 years, a couple of students have put in a lot of work and surveyed the entire landscape for certain species of birds. And to summarize what they have found, what they find is that as you go from the wooded habitat to transformed habitats, which is these invaded habitats, some of these forest birds, forest specialists, seem to actually occur all the way into these invasive new habitats. It's only the grassland birds that have declined in this habitat. So now you have to try and remember that I told you in the previous part that the forest birds were all stable, that their population sizes did not change. But I'm giving you an example here for contemporary time where we've gone and planted some invasive species, and forest birds have kind of exploded even up to 70% into that landscape. And we have not recovered this in PSMC with that particular method. And that's where Rais, our BSMS student, came in, and we started using this another new tool which lets us estimate population demography in more recent times, okay? And this is with one species that we know quite a lot about the ecology, the Sholicola or the shortwing, the species that I studied quite a lot. And we know the landscape quite well now. So this is a Shola, what we imagine it to be, forests and grasslands. But if you've been to Ooty or some place like that, you know that large parts of the landscape have changed. Humans have kind of taken over the landscape. So any would have decreased in this. There's timber plantations, and I told you that birds are there, so any would increase there. And tea plantations, again, any would decrease because you would have reduced the land, the land available. So these three configurations for this one species, I'll quickly tell you. High wavies, where the forest is kind of cut like a cake. One side has tea estates, and what we find is that there is a decline in recent time. Here the x-axis is kind of calibrated to time, so you can read the, sorry about that, but you can see that there is a decline, and then Kodaikanal, where you have both establishment of a big city, and you also have invasive species coming in the 1970s. And very interestingly, we are able to recover both the human habitation led decline and an increase with the timber plantation. So this was really, I was really thrilled to see this. But actually the biggest surprise was this part of the landscape, Berijam, which is near Kodaikanal, used to be a beautiful grassland in the 1970s, and now it's fully wooded. And you can see that the NE is actually increased dramatically over this time. So we are actually able to kind of recover with genomic tools some of the landscape changes and our ecological data that we're collecting on the ground. This data is quite fresh, just a few weeks old, and I'm quite excited about it. And I just want to say that the way ahead is to actually expand this data to other species and geographies, and especially link this ecology and evolutionary processes together. And just to give you a brief glimpse, we've now started collecting data across India. Many species show that different populations are kind of synchronous, but in some others, different populations of a species seem to show different trajectories. perhaps they are responding differently to climate. And that is very informative because when you have some species responding in a particular way and some others, I mean, it might give you some more insights into what a species is adapted to and so on. So in summary, essentially, I talked to you about the Shola Sky Islands, and I like to kind of think of this as this forest grassland mosaic where when one increases, the other kind of decreases and so on. But in essence, what I've shown you is that birds are actually responding not just to the habitat structure, but to the climate. Yeah, and and we've been able to kind of recover some of this demography and demographic changes with genomics, which I'm quite excited by. And I hope that this is going to throw up a lot more questions, especially with comparative genomics. So yeah, thanks. I'd like to kind of open it out to questions if you have any.
Very interesting, I'm sure. Yes, Parth.
So I'll make a few observations and some comments. Number one, the Whittaker plot always seemed to me to be too artificial. Habitats are continuous and it's very discrete. What Whittaker plot comes up with is a very discrete habitat. That's a comment. The second is that you're talking about these specialists and generalists which are found over widespread habitats. You probably, now that you have genomic data, I suspect that if you look at these specialists, you would find the impact of natural selection on the genomes much stronger as opposed to the generalist. And so that would by itself be an interesting thing to look at to relate natural selection, the impact of natural selection on the genomes and their habitat selection. The other point that I also want to make is that as you're looking at these Lee-Durbin plots, there's a major assumption there, one of neutrality. So if there is a huge amount of natural selection on the genomes, these will not give you good estimates of any.
Good point. Yeah, thanks. I mean, I completely agree that there are limitations to what is possible with the PSMC kind of plots, and agree that we need to kind of look at it a little bit deeper, especially the specialists and generalists. We are hoping to do a little bit of that. Thanks for that.
Question?
Hi, Robin. Thanks for that very nice overview. It was intriguing to see, but perhaps expected, that many of these birds are responding to climate, which is a larger biogeographic scale factor, than habitat, which is a small scale. But I was wondering if the results are consistent across, say, different gills. So, for example, if you have frugivores and they have nothing to eat in grasslands, what about frugivores versus insectivores in terms of their response to habitat? Is that something you looked into?
Yeah. So this, the habitat response in general seems to be better at the local scale rather than at the larger scale. Somehow, the climatic responses are better captured at the evolutionary scale. So the way I look at it is that the climate provides the bigger envelope, and then the habitat features come in, and which is why you have these species kind of moving into novel habitats as they are created. As long as it is happening in the same climate space, they are able to kind of move into this newer habitat. So sorry? Maybe refugia. Yeah. So definitely the forest refugia part is definitely there, and it's expanding from there, but over a shorter period than over at the long term.
Just to follow up from that, so she was talking about insectivores versus frugivores. What would you expect omnivores to be? So if you were to look at feeding gills, that might be an interesting way.
So feeding gills in terms of historic demography does not seem to have that much of an impact. That is what I meant to say. But over the shorter time scales, that is where there seems to be some stronger impact. Yeah.
Last question.
Yeah. So Robin, really nice to see all of this coming through after so many years and discussions. Not that it took long, but a lot of discussions. I'm just curious, NEs are very high.
Yeah.
I'm just curious about
why that might be. I'm not entirely sure.
Okay.
I
So
mean,
there's a lot
one
of
thing is
variation
that-
in, but there's not a lot of variation in genetic diversity between species.
So one thing is that we looked for, because we already had a lot of background data, we knew we were looking at habitat, I mean population structure,
Right.
we looked at relatedness, we looked at many of these things, but despite that, this is what we have, this is where we are.
Yeah, yeah.
One of the big problems is to kind of connect any to actual population size,
Right,
which
right.
is,
Yeah.
you know,
But
it's
the-
really,
yeah.
so I have no handle on how these relate.
Sure. The other thing is that basically I don't know, it seems a little counterintuitive because at least from paleontological data, we always expect that generalists with large distributions are more stable, right?
Yeah.
But you're showing that specialists are stable.
Yeah. So I think this is a very, it's a peculiar case of mountaintop specialists, because a lot of the core data also shows that on mountaintops, the climates have been stable and the forests have been stable. So it's not that it's not a generic specialist versus generalist. It's a climate specialist, and it's a climate specialist that survived on these mountaintops. That is what is stable, and which is why we have these birds which are 5 million years, 7 million years old. The oldest lineages of birds are on the mountaintops. They are not anywhere in the lower elevations, because I think the lower elevations got impacted by a lot of fluctuations of climate. So there must have been more extinctions and movement of species from other places and so on.
Great, thank you.
Well, thank you so much, Robin.
Yeah, thanks.
Very good. Okay, so our next speaker is Ulasa from ISER Trivandrum. And now we move to 2 talks on plants.
Good morning. Thank you for the opportunity to speak about my work, our lab's work. So today I'll be talking about plants. I'm not a plant biologist, but Professor Renee wanted me to talk about evolution in the wild, and she would rather have me talk about plants than my butterfly work. This is the first time I'm presenting this work. But I also want to point out, I'm going to talk about this group of plants, impatiens. So Uma talked about birds, mammals, and Robin talked about birds. Very few endemic species in India. This genus, impatiens, has 120 odd species. 90% of them are endemic. So this is in the Western Ghats, and 90% of them are endemic to the Western Ghats. So there's a lot of speciation happening in the Western Ghats. So this gives us a nice contrast with the previous talks. Right. So some of the work was done in collaboration with Dr. Deepak Barua in ISER Pune, but primarily this is a work done by 2 of my PhD students, Sumaya and Sneha. In a sense, I'm compiling all of their PhD work into a few slides here. One of the big questions in biology is why we have so many species. A one-word answer is evolution, or you could say speciation. Speciation is the process, the evolutionary process through which we get more, through which we get new species. Right? So we are broadly interested in speciation in our lab. We also work on other things like color pattern evolution, co-evolution of butterflies with their host plants, and so on. But today I'll be talking about this work on impatiens plants. Right. Just so that we are all on the same page, a species can be defined as a set of individuals that can interbreed or potentially interbreed. So two species are different because the individuals of these species cannot interbreed with each other. Right. So, for example, take a horse and a donkey. They can mate, but this gives you a mule. But the mule, this hybrid, is sterile. This cannot reproduce again. So therefore, the horse and donkey are distinct, even though they can produce hybrids. In a lot of cases, in fact, in most cases, hybrids are not even produced. So you don't normally get interspecific, that is cross-species hybrids. For example, these two birds here have different song, and this keeps them reproductively isolated. So speciation begins as divergences between populations. So we can think of an ancestral species here. So this, you can imagine this as having two populations or two sets of populations. They start diverging ecologically. So you start getting incipient species, which are incompletely reproductively isolated. Over the course of time, this could take perhaps millions of years or tens of thousands of years. Over the course of time, they eventually diverge enough such that they are now reproductively isolated. So this is what we call speciation. So once speciation is complete, sometimes you can get hybrid formation, that is hybridization. And so this hybrid has a mix of gene pools from the two parental species. And if this hybrid is fertile, in other words, it can reproduce with one of the parents, in this case, this parent. So what has happened is genes from this parent have gone to the hybrid, and from the hybrid into the other species. So this is what we call introgression. Right? So once speciation is complete, if you have a lot of hybridization and introgression, potentially the species can merge back. The two species which were distinct can now merge back into the same species, potentially. Right? So in the initial stages of speciation, the probability of speciation is given by two opposing forces. One is differentiation. This is ecological divergence between the populations. And then you have gene flow. So these populations are still compatible with each other. So there is gene flow. So the divergence is counteracted by gene flow. For speciation to happen, the divergence needs to be higher. Right? And once speciation is complete, the probability of maintenance of species as distinct species is given by these two opposing forces. One is ecological divergence, or at least the divergence, if not increase, it should be maintained. And there's homogenization because of introgression. Right? If you have too much homogenization through introgression, the species will not remain as distinct species. So in my talk, I'll talk about these two aspects. I'll talk about these two opposing forces in one set of projects, and I'll talk about the role of introgression in another project. So if you think in terms of geography, you can have multiple ways in which speciation proceeds, but one common, easy to think of way is what is called as allopatric speciation. You can think of an ancestral species which was distributed in a given area. A barrier comes up. This could be a physical barrier like a mountain range or a river. And this barrier reduces gene flow between these two populations. And eventually these two can diverge into new species. So this is allopatric speciation. For example, the Colorado River in North America, once it was formed, it created a barrier, and it created the Grand Canyon. Now you have populations that are isolated on either side of the river, and it's thought that this led to speciation of these squirrels. Now we have two squirrels. One is found only in the north, one only in the south, and this is an example of allopatric speciation. This, you can even date this like using techniques what Robin has presented. So this seems to have happened about 10,000 years ago. In the Western Ghats, we have these sky areas that Robin very nicely presented. So what Robin talked about, they're primarily in the central and southern Western Ghats. In the northern Western Ghats, so this is in Maharashtra primarily, you have these kinds of habitats. So you have these flat-topped mountains. So this is rocky. These are very rocky. So these are rocky plateaus. And you have valleys in between them. So these are essentially sky islands. Right? Now, on the top, it's a very, very harsh environment. In the dry season, very, very dry, very hot. Very few organisms can survive. During the monsoons, there's no soil to seep out, right? For water to seep through. It gets waterlogged. And again, it's a very harsh environment. So plants and animals that live there need to be able to deal with these two harsh environments. Right? And very few plants and animals can survive on these plateaus. So many of them that survive, they tend to be specialists. And one of them is this plant, which I'll just present. So this plant is, if you go to one of these plateaus in Maharashtra near Satara, and almost the entire plateau is covered by a bed of these plants. Very spectacular. And this happens just after the monsoons. This plant flowers, fruits, and the seeds fall down. By that time, it's too dry. The seeds germinate the next year. Right? When you go closer, they look like this, and these are the flowers. So this is a rocky plateau specialist. It's found nowhere else. And what really caught my attention was this. So this is Khas Plateau, where it looks like this. 17 kilometers away, a different plateau here called Chalkewadi. The flowers look completely different, right? To me, they look like two different species. It's not just the size, the shape, the color. This is deep pink. This is whitish, right? This is Toshegar. This is intermediate, 8 kilometers from here, 15 from here. The flowers are intermediate, right? So keep in mind, this is not the kind of scale Robin works on. This is a very, very small geographic scale, right? So this is what caught my attention, and I wanted to see what's going on, right? So a simple question. The strong specialization to rocky plateaus, is this driving allopatric divergences, right? Potentially leading to incipient species in this system. Right? This is impatiens loviae. So this is what we studied. So as a control, as a comparison, we included another impatient species, Impatiens oppositifolia. This is found on the rocky plateaus, but also in the valley. So it has a continuous distribution. So this serves as a nice control, right? A simple hypothesis. There should be stronger allopatric divergences in the plateau specialist compared to that in the generalist, that is oppositifolia, right? So this was very hard work going to the plateaus. Some of these are remote. Reaching there itself is a task, right? Students have quantified a lot of different this is flower traits, but they also quantified other traits. So most flower traits are positively correlated with each other, which means they vary in a correlated fashion. So it's not just one or two traits that differ between the plateaus, but there are multiple floral traits. So here we have a representation of the differentiation within Loviae. The three colors represent three plateaus. And you can see that the plateaus cluster out quite well. But in oppositifolia, which is a generalist, there seems to be no differentiation. Right? as we predicted. This can be represented in another form. So here you have multiple traits combined here. You can see that the three plateaus are very well differentiated. This is impatiens loviae, which is a specialist, but in the generalist, there's no differentiation at all. Now, why do we have this differentiation? Any phenotype you take, the phenotype, you can have the effect of the genotype, that is variation in, if you have variation in phenotype, that could be because of variation in the alleles or the gene composition, or it could be because of the effect of the environment. For example, plants, their height, the branching pattern and so on can depend on the light conditions. Here's another example. I digress now towards butterflies. So this butterfly species has green and brown pupae, the cocoons, right? And there's a tendency for green ones to be found on green leaves and brown ones to be found on soil, rock, and so on. So it seems like where the pupate matters, right? And we wanted to know whether this is phenotypic plasticity. We've done a lot of experiments. So in one experiment, we can take these larvae, the caterpillars which are about to pupate, put them in these containers, green and brown, and here we have data on the color of the pupae. So higher values indicate more green. So if we force them to pupate on brown containers, they tend to be more brown. On green containers, the pupae tend to be green, right? So this demonstrates that pupil color is phenotypically plastic, right? Now, we could do this because we had a hypothesis about green versus brown, but with the impatiens loviae, you can't do it because we don't really know what may drive, what environmental factor may affect their phenotype. So what we did was something in a way opposite to this. We brought all of them to the lab, and we grew them in what is called the common garden experiment, right? So here we have this differentiation in the wild. So now we bring them to the lab, grow them in greenhouse conditions. Now we ask, are these differences also seen in the lab, or do you lose these differences? If it's phenotypic plasticity that's giving you this variation in the wild, under greenhouse conditions, this variation should be lost, right? But if the plateaus are genetically different, if they're genetically differentiated, the variation, the differences between flowers should be preserved in the greenhouse conditions as well, right? So these were the experiments. So we took seedlings, so this is before they flower, brought them to the lab, grew them in the lab. This is what we see. This is data from the wild. This is for oppositifolia. This is loviae. Here you have data in the transplant elements. You can see overall the patterns are preserved, right? The differences are also there. This suggests that the plateaus are, the populations in these plateaus are genetically differentiated, right? And we also did this experiment with seeds. We collect the seeds, store them for 1 year. In the next year, we germinate them and grow them in the lab, in the greenhouse. Again, you see the same differentiation. Absolutely no differentiation in oppositifolia. Right? So it seems like the plateaus are genetically differentiated. Keep in mind that the plateaus are so close to each other, just 15, 20 kilometers. Still, they're genetically divergent, right? Very strong divergences. Now, we did the same with vegetative traits. Vegetative traits are the non-flower traits, height, branching pattern, leaf size, and so on. So I won't go into the details, but I want to point out that the variation is inconsistent. You don't get the same patterns that you see in the flowers, right? So then we asked what's special about flowers, right? So this divergence seems to be primarily in flowers, this genetic differentiation. So are the pollinator communities different? Are they selecting for different flower colors, right? We took 2 plateaus. We looked at the pollinator visitation, all insects that come. So these are the raw data, so to say, and these are scaled in terms of proportion. You don't need to go into the detail, but all I can say is that, yes, there are differences between the plateaus we studied, but it does not seem like the differences are strong enough to to explain the kind of variation that we saw in the greenhouse experiments and in the wild. Now, if you look at this, Chalkavadi and Toshegarh are close to each other. Khas is far away, well, relatively more further away. But in terms of the morphology, Khas and Toshegarh are very similar. Chalkavadi is the most divergent one. And this is also reflected in the principal components analysis. Chalkavadi clusters out, right? So there's something happening there. It's not just geographic. It seems like it's not just geographic distance. So we want to look at this. We did a more comprehensive survey. So we went to a lot of different plateaus and tried to identify all populations of impatiens loviae, right? Not all plateaus have them. They're found only in some plateaus for some reason. And so we identified 7 plateaus which range across the entire north-south distribution. And again, you find the same pattern. In the specialist, the plateaus separate out quite well, whereas the generalist does not separate out. This again tells us that habitat specialization is driving divergences between populations. And these are further analysis which point out to the same pattern. Here you have, this is giving you some idea of morphological differentiation across plateaus. It's much stronger in loviae compared to that in Oppositifolia. We also looked at the population genetic structure using AFLP data. Again, you have the same pattern. In loviae, you have strong differentiation. In Oppositifolia, the control, the generalist, it's not so strong, right? And so now, we wanted to see what's the role of genetic drift versus natural selection, right? So genetic drift is simply differentiation between, so in this context, it is differentiation between populations by random chance, okay? Or you could have differentiation driven by natural selection, right? I'm simplifying this a bit, but it works for this presentation. Now, if you have genetic drift, what you expect is that they should be, as the differences, as the geographic distance between population increases, the genetic distance should also increase. Right? So that's a very straightforward prediction. So you should have a correlation. You could do it with a morphological or genetic distance. And the prediction is that this relationship will be steeper in the specialist, and it's either absent or weak in the case of the, so it should be steeper for the specialist and weaker for the generalist, right? So morphological distance versus geographic distance, there's no trend at all. Genetic distance, yes, there is some signal for isolation by distance, suggesting that genetic drift does explain some of the variation that we see. But for morphological distance, for some reason, we don't have that, right? Then we looked at natural selection. I already talked about the pollinator data. The pollinators don't seem to be different enough to explain the patterns. And we looked at the effect of the climatic variables. And a simple, straightforward prediction is that population with more similar climatic regimes should be those that have lesser genetic distance. Right? For morphological distance, we looked at multiple climatic variables. There's no correlation with the geographic distance. But when we look at the genetic data, there are a few variables. For example, precipitation data can explain this, some of the genetic differences that we see across populations. To wrap this up, strong specialization seems to drive allopathic divergences across populations, and this can be explained both by genetic drift and environmental selection. Now, very quickly, I'll talk about the second part. This is about introgression. So in this species, Impatiens balsamina, so this species is found, as you drive along, it's found along the roads as a very dense set of individuals, very common, right? Now, there's one more species called Impatiens rosea. So Impatiens balsamina has a very long nectar spur. The nectar spur is where nectar is stored. So pollinators have to land here, and they have to insert the proboscis or the tongues to reach the nectar spurs. Therefore, the pollinators for this species are likely to be different from the pollinators for this species, which has a very small nectar spur. Right? So in fact, Darwin saw these flowers, an orchid which was presented to him, it was gifted to him, and he predicted there should be a moth which has a tongue, a proboscis, which will be able to take the nectar from this plant. So but at that time, no moth was known which had a 30 centimeter long proboscis, but a moth was indeed discovered later on. Right? So here, this is the Deccan Plateau. So here you have the high altitude mountains, the rocky plateaus. You have the Konkan plain along the coast, and you have the Ghats, right? So this is where if you drive along one of the Ghats in the Maharashtra, you climb down or climb up this Ghat section, right? So the Ghats look something like this. So we found Balsamina, when we were surveying this for some other project, we found Balsamina only along the Konkan, the coastal plain, Rosea only in the tops, at least in the area where we were working. Interestingly, along the Ghats, we found hybrids which had intermediate sized nectar spurs. So this, we didn't expect this to be there, but we found this. And then we went up and down multiple times, and we found it very surprising. You start at the top, you find only Balsamina, very, very abundant. So you find only Rosea, very, very abundant. You drive through the Ghats, you suddenly see hybrids, only hybrids. You reach the plains, you see only Balsamina, right? And what was even more surprising, this is at the top, you have only Rosea. So here, this is where you have the Ghats. So we found the hybrids, just a few kilometers, very narrow, and then you have Balsamina. Keep driving, again you get the hybrids. This is all along the lower elevation. A few kilometers away, again you get Rosea, right? Very weird. In fact, in this river, this is Savitri River, on the northern side, you drive along the coast, you get Rosea. After a little while, you get Balsamina, again you get Rosea. In the southern one, you have a different species. So we wanted to understand what's happening here. So first, we wanted to confirm whether these are actual hybrids. So we did morphometric analysis. So it's not just a nectar spur, there are many other traits which we included. Yes, the hybrids cluster out between Rosea and Balsamina. We brought them back to the lab and did cross-pollination experiments. So you take pollen from one parental species and drop them into the other species and so on. And here you have, again, the same pattern. So the lab hybrids, the lab population, they're in squares. The wild ones are in circles. You can see, again, all of the lab hybrids cluster out with the wild hybrids, which tells that, yes, there can be hybridization, and you can replicate that in the lab. We wanted to understand why is there this pattern? How are the species separated out, right? So this is a climatic niche model. To summarize this very quickly, you can look at the color patterns, and you can see that all of them have the same climate suitability. It's not that one is more suitable, so to say, for the higher altitude and lower altitude. Their climatic niche, the climatic niche is the same. So there is more or less complete overlap. That does not explain why do you have this geographic patterns in the wild, right? So I'll skip this part. And now I'll come to this more interesting. Why are the hybrids so narrow? Why is the hybrid zone so narrow? Why are the populations not mixing up entirely? Why, right? That's a question we wanted to ask. Right? One possibility is that the hybrids have very low fitness. Right? You do get hybrids, but they have low fitness. They have low survival or reproductive fitness. Again, we did these analysis in the lab. You can take pollen from one, create hybrids, and so on. So we have a lot of data, but I will just breeze through them. So here we have one estimate of fitness. This is seeds per fruit. Here you have the self-pollination treatment. That is, so this is for impatiens Balsamina. You take pollen from Balsamina and place it on Balsamina. You have cross-pollination, which is where you get hybrids between two species between Balsamina and rosea and so on. So now you see that there's no differentiation. The hybrids don't really have lower fitness. And again, here you have germination rate. This is seed germination percentage. There's no difference between the hybrids and other treatments. We looked at many different traits. Hybrids do just as well as the parents, at least under lab conditions, right? Many other things we have looked at. Pollinator visitation in the wild, we don't find much difference. Surprisingly, even though the nectar spurs are so different, they have the same pollinator communities. These are at least the diurnal, the day active ones. We have not looked at the night pollination, right? Then we wanted to check whether there's a difference in adaptation. Balsamina may, for some reason, be better. It may have higher fitness in its native range compared to that in the foreign range, right? If you take Balsamina and grow it in the rosea's range, what happens, right? So we did these experiments, we did these transplant experiments. We take plants from the hybrid zone, transplant them to the Balsamina rosea zone. You take the rosea ones, transplant them to the Balsamina zone, and so on. We have a lot of data, but I just present a couple of slides. Here, for example, you have survival. The red curves indicate survival in the foreign zone. The cyan ones are survival in the native zone. So you see here, for example, survival is a little better in the foreign zone. Here it's a little better in the native zone, and so on. So there are some differences, statistically significant differences, but there's no strong pattern. What it means is that they survive just as well. They have the same fitness, irrespective of where they grow. And the question is, why hybrid zone so narrow? Why is it so narrow? Just a few kilometers. Why is it not getting mixed up, right? I don't have a good answer to this, but we have some data from the population genetic analysis. So we've done DD-RAT sequencing. What we've done is we've taken a transect. So this is one hybrid zone. So from Mulchi, this is rosea. This is Mulchi hybrids. You have the Balsamina. Then you have Dapoli hybrids and rosea, right? So the Mulchi rosea, and okay, this is not intended, but anyway, the Mulchi rosea is here. The Dapoli rosea is here. What can I do? Okay, the magic fingers worked for it, right? So they are the most distant, geographically distant, but they belong to the same species, right? Here is a table of pairwise FST values. FST is simply something that, I need to wrap up. FST is something that tells you how different the populations are genetically, right? So higher values indicate more genetic distance. I just want to point out that the highest genetic distance was between Mulchi rosea and Dapoli rosea. So the same species, two populations, they have the highest differentiation compared to everything else, right? Very, very strange. And then this is also reflected here. If you, each column here is one individual, and the color represents its ancestry, genetic, determined by the genetic composition, right? So the Dapoli rosea has a completely different color, which means its ancestry, it's a different genetic cluster, right? The Mulchi rosea is different. Then you have Balsamina, which is represented by pink and so on. Without going into much detail, it seems like I rosea, the two populations are genetically the most distant. And we also seem to have some suggestion of introgression. And this is asymmetric. Introgression is more from Balsamina into rosea than the other way around. So what we think is that the hybrid zone is dynamic, right? It's recently formed, right? And the Balsamina may be invading into rosea. Balsamina may be pushing rosea, making it smaller and smaller. If you follow this over multiple decades, perhaps we'll see this change happening, right? Yes, thank you. I think I overshot by a minute. Yeah, thank you.
Just very quickly, you now have genetic data. Have you estimated genetic diversity in each of these? And do you find any evidence of bottlenecks? Could there have been bottleneck effects? Because that actually can completely obliterate the relationship between genes and geography?
So it's a very good question. We've not looked at the bottlenecks. We do plan to do those. But one thing our genetic data do suggest is that there's very high inbreeding, right? So these are called impatiens possibly because the fruits, they're not dispersed. The primary mode of dispersal is not through birds, but they burst open. And the seeds, they don't disperse too far. So maybe there's very high inbreeding, which keeps the hybrid zone very narrow. That's potentially what maybe what's happening here. Yeah.
Okay, thank you. You'll have to catch Ulasa for other questions during the tea break. Thank you.
Can I ask one question?
Oh, okay. Very quick.
I think, yeah, impatiens is a very complex genus, and we know that. And I think that is the best model for this study of evolutionary studies. So I was thinking that when you took those morphological, when the genetic distance was very low in case of the morphological analysis in comparison to genetic distance. So that was very less. So could be, I think some of the parameters might be missed. Say for example, the calyx spur and spurless calyx. So I think there is hope to really investigate deeper so that you can get that higher distance also.
Certainly. We've looked at what traits that we could look at.
Yeah.
But I must point out that there's a weak correlation between morphological distance and genetic distance. So there is a correlation. Plateaus that are more genetically distant are also morphologically more distant.
But state live is a good model, certainly. You approved it. Thank you.
Okay, thank you, Ulasa. Right, so now we come to the final talk of this session, which will be by Meghna Krishnadas from NCBS. And it'll be another talk on plants.
Yeah. Am I audible? Okay, right. Thank you. All right. Good morning, everyone, and thank you so much. It's a pleasure to have this opportunity to present my work here at the Academy. And what I'm going to present is going to have a slightly different flavor. I was from the talks that you have seen so far, in that you're not going to see a lot of genetic data or a lot of molecular data. This is something that we are sort of considering getting into. But I am by training and by a lot of my work historically has been that of community ecology. So which means that instead of, I sort of look at patterns and processes at the scale of the here and now. I look at what is governing how communities are assembled and how different species come together to form a community and stay there. So the way I look at it is many of the evolutionary outcomes that we see at, if you're looking at deep time or larger spatial scales, things that we broadly characterize as niche differences or, speciation that has taken place. The way I see my work is I look at what is the ecological stage on which many of these evolutionary processes seem to play out, and can we look at what's happening here and now in terms of what is shaping these many species coming together to form a community. And so that's the kind of flavor in which you'll see much of what I'm presenting today, but also historically what I have done. Now, I have worked mostly in the Western Ghats. That's where I have a long-term project going on, and that's where a lot of my work has looked into the diverse communities of trees that we find there, and how do we explain so much tree diversity and what maintains this diversity, both in terms of the response of plants to their abiotic environment, but also in terms of the biotic interactions that they have with, say, insects and pathogens. And this is not work that I will present today, but this has been the broad focus of what's been going on in the lab. Now, because of the situation or the geographic situation in which we are, this event is being held, which is IISER Tirupati, I thought it would be nice to present some of the newer work in the lab that we have started in the Eastern Ghats. And this is looking at how plant communities are assembled and what seems to be happening to plant communities in the surroundings here that we see today. And this is something that this is what I will focus on in terms of my talk today. Now, Ecosystems across the globe are facing a myriad of changes, and a lot of this is because of human activities. So humans, in their quest for space, resources, have altered ecological systems at large spatial scales, which compare with that of what meteorites and volcanoes have done in the past, in some sense. And this has led to issues such as fragmentation and degradation of habitats, large-scale nutrient deposition, species invading into habitats where they've not been seen before, and of course the looming specter of the climate undergoing some massive changes. And these global environmental changes, individually and interactively, are reshaping ecological communities at massive scales. Now, to understand how these communities are going to respond to these changes that are happening fairly rapidly, today more than ever, we need to first understand how do these ecological communities form, come together, and stay in the first place. So what is it that governs where, how these many species assemble, and therefore can we then predict how they might disassemble or reassemble in terms of what happens with ongoing changes to the situation where they find them. Now, biotic assemblages vary in we know that they vary in space and time, and this comes about because of certain processes. You may have evolutionary processes creating a meta-community or a historical pool from which species sort of have to disperse into these various smaller spaces where we eventually find them and study them as ecological communities. And even after they disperse, in order to establish at a place, they have to go through an environmental filter, which is that they need to be suitable to these particular environmental conditions. And you saw some reflection of that in terms of what Robin was saying, what is a climate specialization or habitat affiliation. So not all species can be found everywhere. And once they do come to a point where the climate is suitable for them, they still have to contend with other species in that milieu. They need to have they engage in these biotic interactions, which can be competition with species in the same trophic level, or interactions such as pollination, herbivory, and those which are occurring with other species, dispersal, which allow them to then stay in the community and form these diverse communities, as it were. Right? So the broader theme of my research or the research in the lab is to understand some of these processes. Like when we look at ecological communities in nature, when we look at multiple species, can we understand which of these processes seems to be largely shaping these communities, and then do smaller scale experiments to tease apart some of these processes. But the idea is, can we then help detect or use this understanding to help detect how biodiversity may change or across space and time with ongoing pressures that they are facing. Now, the reason species are not all species are found everywhere, and species seem to prefer one place over the other, is because they differ in their inherent features. And this is known as traits. These are morphological, anatomical, or physiological characteristics in which govern functional responses of species. It might be how they respond to temperature or water availability, nutrients in terms of plants, right? So how they use resources, how well they tolerate stresses, or how well they can compete with each other or have certain interactions that they engage in. But the thing is, the role of a trait of any particular functional feature can vary with the context in which you look at it and the scale at which you look at it. The same for plants, for example, there is a trait which is called specific leaf area, which is the amount of leaf area per unit gram of leaf tissue. And it's considered to be a really important biogeographic trait in terms of how specific leaf area is distributed at different parts of the world. However, the role of specific leaf area may be very different when you have plants that are in the Western Ghats, which is a fairly wet, humid ecosystem, versus the Eastern Ghats, where it has to deal with a different kind of stress. The same value of the leaf area might be there, but they might be playing different roles in the way they are filtering species in these two different, very different ecosystems, right? And in this context of trait-mediated assembly of communities and trait-mediated understanding of ecosystems, surprisingly, dry ecosystems across the world, and particularly in the tropics, have been undersampled and underrepresented. So a lot of our understanding of how traits or functional features are shaping communities is based from only a subset of the kinds of communities or the kinds of biomes and climatic conditions that we see around the world. And this is a gap because many of these tropical dry ecosystems are actually under severe threat. Many of these species are considered, they're already very harsh climates, they have really strong dry seasons, and many of these species are considered to be at the edge of their ability to tolerate some of these conditions. So today more than ever, we need to know how traits are determining what's happening in these understudied systems, right? And we know that for these tropical dry systems, the species have to be uniquely adapted to withstand these conditions that I mentioned, right? And even within this fairly harsh system, we know, when we start zooming in, we know that there is environmental heterogeneity. And we know that species differ in where they are found. So there is some knowledge that there is environmental filtering happening even at this scale, but what is it that's driving these filters, and how do the traits mediate some of these changes to the communities is still not very well known for some of these systems. So this is the broad context in which we started our work in the Eastern Ghats. This is work that I started while I was in CCMB Hyderabad, and we thought that we would take this fairly large spatial scale of 6,000 square kilometers encompassing 3 protected areas in the Eastern Ghats. This is a forest to savanna transition that happens from the south here to the north, which the southern parts are Gudla Brahmeshwaram Wildlife Sanctuary, which is relatively more moist, and as you go north, it becomes much drier and more open savanna. But there was a fairly short gradient of temperature and precipitation, but a lot of variation in terms of topography, and which led to variation in terms of like hills, valleys, and slope, which led to microclimatic or microenvironmental variation that we considered would be important. And the first thing we wanted to see is, does woody cover and tree species composition change across this system? And we did that laying out these many vegetation plots across this very large landscape to characterize what the tree community looks like here, right? And when these sites, as expected, differed in their climatic conditions, we looked at the macroclimate, which is here. We pulled this data from remote sensing, from publicly available databases that are there, and we found that the sites that we studied could be discriminated by sites that had that were warmer, slightly warmer, but surprisingly wetter, but had stronger temperature seasonality, which was the this was the set of variables that were most strongly discriminating the sites, right? But we also had microenvironment, which seemed to play a pretty strong role, but not as much as the macroclimate, in terms of discriminating sites with moist soils and high phosphorus, and some sites which had low nitrogen but were acidic in nature. Now, what I'd just like you to note here is that overall, even though the macroclimate gradients were really short, actually it explained a lot more variation among the sites than microclimate gradients wherein there seems to be a lot more heterogeneity. And this is something, the significance of this is something we realized when we started looking at what was happening to the communities that assembled along these gradients. And in short, it seems like macroclimate did not seem to matter so much for the diversity of tree communities that you would find. And maybe that's not surprising. Like I said, the macroclimate gradient, although sites differ, it's still a pretty short gradient. What seemed to matter the most is that there was microclimate, and particularly soil moisture, that seemed to play a role in structuring the diversity of communities across the system. But surprisingly, usually you would expect that wetter sites, and this is what we see at a macroecological scale, wetter sites have higher diversity. We found the exact opposite pattern here, which is that wherever there was higher soil moisture in the dry season, we surprisingly had lower diversity. And most of these really low moisture sites were where we had much higher diversity, which sort of led us to believe that maybe it's because in this overall harsh macro environmental condition, most species are adapted to dry conditions, and that's where you find most of the large communities. There are very few species that are actually adapted to being able to survive in wet sites, and these are probably remnants from historical remnants because of evolutionary refugia. And some of this is indicated in the functional diversity, which tells us how these communities are assembled. So if you factor out what the species richness is and just look at how much diversity there is in traits, we find the exact opposite pattern, surprisingly, and we find that these drier communities are comprised of species that are very similar to one another in their ecological features, in their traits. Whereas in the wetter sites, you find that there is more scope or more leeway for traits to play out, right? Which also seems to suggest that, you know, some of these species might just be these remnant wet associated species that are there in these wet refugia, likely the valleys that are there in this otherwise very dry landscape. Okay. But we did see that, you know, precipitation and temperature led to how species, you know, the change in species as you go from one site to the other. So let me just walk you through this. The change in species or the similarity of ecological communities or the dissimilarity of ecological communities here can be characterized as something called an ecological distance. That is, we ran a model where we said that, okay, if there is this much difference in the community of trees that are found from one site to the other, could you put that together as a predicted, a statistically significant ecological distance between these communities? And then you check to what extent this ecological distance relates to the change in, say, a variable like precipitation or maximum temperature of the warmest month. And how you read this is that as you go from a precipitation of 90 to 95, you don't have a lot of difference in what the community looks like. But when you go from 95 to 100, the community starts looking very different, right? Which suggests to us that at some changes in the temperature level, you're going to see big shifts in what species are going to be found, and that comprise many of these communities at these large geographical scales. But I'd like to draw your attention to one thing. If you look at the numbers here, the predicted ecological distance, which we found, this is actually in the data, right? And you look at what is the partial ecological distance that is factoring out all other variables, what is the ecological distance attributable to this variable alone. Macroclimate again seemed to explain, even though this curve looks very steep, the actual ecological distance or the dissimilarity in communities with respect to macroclimate is pretty small. And again, when we compare that to the microenvironment, primarily soil moisture again, but also soil phosphorus to some extent, which if you recall were the two variables that loaded the most dominantly onto that first principal component axis that was characterizing the microenvironment differences. We find that a much larger amount of the ecological distance, even after factoring out all other variables that are possibly driving it, is being explained by soil moisture, which gives you the idea that dry season soil moisture seems to be a very critical variable in this system at a scale which is telling you how communities are going to change as you go from one place to the other across this system. And some of these changes were driven by the response of individual species. Whatever I presented so far was this composite picture of what's happening to the community as a whole, right? But we wanted to get down to the base levels of, can this be explained by what different species are doing? Because eventually when we measure traits, we are going to measure that the phenotype at the level of species, and what we assume that species are getting discriminated based on their functional responses that are that traits somehow represent, right? And yes, we found that if you take the macro environment, their species clearly differ in their response, which is that there are some species, these values here, these species like warmer, wetter environments, but are also able to withstand stronger seasonality of temperatures. A bunch of species that don't seem to care much, and this is interesting because you have many, many species here which seem to be generalists, which is sort of what we expected, and which is also what would sort of tell us why is it that you have a very low functional diversity in many of these sites that we are looking at. But we do have some species here which seem to prefer, go in the opposite direction and prefer conditions that are cooler but can be drier. And this was something surprising and something that we would like to go into further, like what are these features that are discriminating species along these unexpected dimensions of temperatures and precipitation combinations, right? But again, the microenvironment seemed to be a much stronger discriminator of species responses across this habitat. What we found was there was a whole bunch of species that seemed to prefer moist soils and high phosphorus conditions, and many of these species, these genera that we know, are associated with riparian valley vegetation and things that have congenerics in the Western Ghats, which is, wetter climate, and many of the species that we found here that were preferring the more dry sites were actually those that to be very dry associated species found in the Eastern Ghats and habitats around here, right? So again, we find that species are certainly responding to moisture and nutrient conditions, and primarily moisture. A lot of this was, we know that, moisture was the main driver of many of these responses. But what we found was the traits that we chose to study were very poor predictors of these species' responses to the gradients. So even though we know that there was clear discrimination or differentiation among species in how they were reacting or responding to these gradients across the system, traits did a very poor job of predicting these responses, or at least the traits we studied. And that's why we found that even though there was compositional turnover in terms of the species, the dissimilarity models that I showed, again, the compositional turnover was not predicted by the traits that we chose in this system. And one reason could be that the gradients are too short, but we do see that there are clear differences among species in what they do. So the question is, what is going on here? It is, what is it about the species that might be able to explain this? The first thing you would think of is that, as I said, these traits are not really the traits relevant to the key drivers. So for example, if temperature is the key driver of where species are found, would thermotolerance or drought tolerance be a trait that we should be looking at? And these higher level, more synthetic traits, such as those of, the traits we studied are basically specific leaf area, dry matter content, bark thickness, and wood density. And these are traits that are more integrative. They are more at a whole organism level, and maybe they are not the ones that are really driving these mechanistic responses of species. However, when we did these experiments, we took some of these species, which are 15 of these species from this large study that we did across 48 odd species, and we said, let's choose species that are found only in dry sites or drier sites, slightly wetter sites, and the ones that are more broadly distributed. This is from the graph that I showed you for how species are doing different things, right? And then we said, okay, let's measure these simple traits, but also a few more complex traits like what we call Turger loss point, which is a very good indicator of species response or their drought tolerance, right? And here with Turger loss point, what we would find is that when the values here would suggest that these are more less drought tolerant. And surprisingly, what we find is that by and large, when you are more drought tolerant among all of these species, you actually tend to accumulate better biomass, which means that yes, all of these species in this dry system would probably, the species more accustomed to drier conditions are going to perform better in general. But when you impose a drought treatment, which is this red line here, the difference among species in their roles or in their ability to accumulate biomass doesn't really change. So which means that Turger loss point as a whole, like macroecologically, is driving what's, how species have come to be in this environment, but it's not discriminating species' response to water, a further loss of moisture from what we see. Surprisingly, leaf dry matter content, which is a simple trait, is still shaping or explaining how much biomass you're going to accumulate in dry versus wet conditions, where low leaf dry matter species, which are thought to be less stress tolerant and they are photosynthesizing more rapidly, they are actually, they as expected do well when there is plenty of water available, but their performance dips when there is drought, right? So clearly traits do matter at the scale of these experiments we do, but they don't seem to then translate into explaining what's happening at the macroecological scale of how these communities are assembled, right? And one reason that might be is because there is immense variation within species. And I seem to have taken a lot of time, but this is something that we've been excited about because this is fairly new from these systems, that if you have a system that is fairly homogeneous environmentally, do traits need to vary? And the answer is yes. We see that some of these traits, bark thickness, LDMC, leaf matter per unit area, and wood density, some of them have more variation within a species than they have among species, right? So in some cases like wood density, highly conserved trait, we don't see it, this was expected. But some of these other traits vary a lot, which might explain why we don't see this kind of, we don't know whether this is because of genotypic differences or because of plasticity in these traits, but this is why some species or many of these species can be found everywhere, right? But then we wanted to understand, is there still some role played by these, by the fact that there is so much variation and the fact that some species tend to vary more than others in their traits, right? Or the variation within species is really high. And what we find is that yes, intraspecific trait variation does relate to ecological properties, and a property such as abundance or the geographical dominance of a species correlates with intraspecific variation. If you have more variation in your phenotype, you tend to be more geographically dominant. This is kind of a consistent response in multiple traits. I won't go into some of the details of these parabolic responses that we see. We also see that when you have more variation in your traits, you tend to occupy wider niches. And this might be one reason that you're occupying many more niches, and that's why you are able to become more abundant. And we do see that if you are occupying more niches, you're more abundant, right? So these are various pieces of the puzzle. But the thing is, does it all come together? Because there are multiple ways that variation in a species trait can link to its eventual geographical properties that are macroecological, right? One is niche width, like I mentioned, that if you have an environmental gradient and you occupy wider niches, you may just be able to persist in more sites. So that increases your plot occupancy, and that increases your abundance. But another thing that matters is how is the environment distributed in this place in the first instance, right? So if you have some species that are very close to the most common environmental conditions, which we term as low niche position. The blue here is showing the distribution of the available environments, or very broadly speaking, available niches, and you're very close to the most available niches. Low niche position, maybe that's how you become more abundant and has nothing to do with niche width, as opposed to something that is occupying very peripheral environmental conditions, right? So we put all of this together in structural equation models in which you can see which one of these factors relates to which one. Is it a direct relation or is it a mediatory relation? And we wanted to check how intraspecific variation goes on to shaping abundance, which is an important property of species, right? Is it through niche width or the niche position? Does that drive how many sites across an available landscape can you occupy? And does that then explain your abundance? And what we found is that two major traits seem to play a key role: bark thickness and wood density. Unsurprising in this landscape where fire is a key driver and bark thickness is a major variable that relates to how you respond to fire. But let me just summarize the key relationship, which is that bark thickness, the trait variation, although we know that it relates to niche width, that does not lead to any or has no mediatory role in governing the eventual macroecological properties such as how many sites you occupy or how abundant you are. What matters is how close are you to the most available conditions that are in a system. And this relationship shows that if you have wide variation, surprisingly, those species are actually occupying conditions that are very close to the environmental mean, and those are the ones that are occupying many more plots and therefore becoming more abundant. So how we are interpreting this is that if you have wide variation, you're going to have, you occupy many wider niches, and that allows you to occupy, to sort of sample much of the landscape. But really what matters is that you're occupying many of the plots by being close or being able to take charge or dominate the most common environmental conditions. And that is somewhere where the next step comes in, like being in these environmental conditions, these common conditions, do you have to vary a lot in trait to actually change how you compete with other species when you're in these conditions, right? Which is something we can't test in these sort of studies, but more detailed analyses of long-term forest dynamics or vegetation dynamics can let us know. But eventually what we see is that you occupy more the conditions that are representative of the most common environment, and that's going to drive how abundant you be, right? So to summarize, soil moisture is a key driver. The dry sites host more diversity, and it seems like most species are very well adapted to these systems. And while there is a very high turnover with soil moisture, we don't see that being related to traits as much, and that's probably because intraspecific variation is really high, and really what is mattering is the phenotypic differences within species that is allowing them to occupies some of these places and determines macroecological properties, right? And so some of this work tells us fundamentally how are species, or what is it that makes species common or rare? You know, can we find these properties that we can measure very simply and predict, is this going to be a common or a rare species? Because commonness and rarity is very closely related to extinction probabilities and how species might respond to changes in different situations, right? Which again, as I come back, is important for us to predict that if this climate or the particular microenvironment in a region is changing in a certain predictable way, can we use these properties of species to then predict how they will respond to these changes that are ongoing, right? A lot of this is very new work, as you saw, and most of this work was led by different master's students who came into the lab. So I'm very grateful to them for all the hard work of working in these very, very tough conditions, doing these plots and generating all of this data. You know, hopefully some of these publications will come forth very soon. And I have to thank, this was from a grant by SERB, the Early Career Grant, and also funded by CCMB when I was there. So I have to thank those institutions and all of the students who did the fantastic work in bringing all of this together. And thank you so much for your attention.
We have time for a couple of questions.
Hi, Meghna. Really nice work. I had a question about whether any of your sites had disturbance gradients and if this affected the community diversity that you found.
Yeah, depends on what you call disturbance, like whether fire is a disturbance or not. In the sense, it is an ecological disturbance, but it has an ecological role. But the reason we chose this landscape is because it has relatively low human footprint, right? So there wasn't lopping, felling, biomass extraction, or things like that.
No grazing also?
No, very little. No, no grazing.
Okay, so
I won't say no, but very little grazing.
And what about invasives? Were they in your plots, or did that play any role?
Great question. We avoided plots with invasives, but then we started a parallel study in which we looked at, and we are still continuing it. There's a long-term effort where we are looking at lantana that has invaded some of these systems, replace the native understory grass, and we are trying to see how this invasive is changing the dynamics of tree recruitment and mortality, and mostly at the younger life stages. And interestingly, there we find that traits seem to predict the response of these simple traits, like leaf traits, seem to predict how species are responding to an invasive lantana.
Okay, absolutely the last question.
Yeah, thank you for a nice talk. I'm a chemist and I was wondering about just one element figuring in your slides, that is phosphorus. And how about nitrogen and other things? Does phosphorus have any role in what you are doing?
Yes, so in many of these, so of course nitrogen and phosphorus are both key, are known to be key nutrients that limit plants in these systems, and by and large they have a macroecological value in terms of how communities are changing. So that's why nitrogen was also there, by the way, and we did have other nutrients as well. We also had pH and carbon nitrogen, but none of them were playing that important a role. It was primarily phosphorus and nitrogen that discriminated plots, and phosphorus from smaller scale individual studies of plants we know imposes the strongest limitation often.
Okay, thank you very much, Meghna, and thank you to all the speakers of this symposium. I think we had a wonderful time, and thank you very much. I think we have a tea break now, and we come back at 12, sharp 12.
Arun Kant, and I am a civil engineer by training, and from IIT Bombay. Now, I have great pleasure in introducing Mr. Dr. Pradeep Kumar Singh, former director of CIMFR Dhanbad, who is going to speak on blasting, I think controlled blasting, and he's going to tell, so it's something which will interest most of us, how to do. I think in Bombay, they use this technique for demolishing tall buildings, old tall buildings if they want. Recently, they did it in Bandra. There was a hotel, and that has been demolished, Okay, let's listen to
Thank you,
Dr.
Chairman,
Singh.
and good afternoon. Yesterday, I got cold in the early morning in the Darshana, so I am sorry for it that I will not be able to speak properly. But this is a work, what we did, it is a work for 7 years, because this Navi Mumbai airport, they planned to have the Navi Mumbai airport, but there was no land available in the periphery of that. Then they invited a committee meeting, and they decided that the Ulve Hill is having around 3,000 acres of land, and that is the only area where a greenfield airport can be constructed. It was a difficult task to construct the airport, because in the hill itself, there were a number of residential people who were illegally residing in them. Illegal basalt mining was going on. But government decided that, okay, we will have a continuous monitoring of scientists and technical staff, and that way it will be constructed. So in 2016, November, we started the work. First blast was conducted there in 2016. And the timeline given by the CIDCO was that all the construction work, this blasting work should be completed by 2023. And I am proudly saying that we completed all the job, because they deployed 4 construction companies, and we submitted the designs, implemented it under supervision, and without any accident, without any fatal, and without any litigation, whole work was executed. So that's briefly how science and technology can be used, and explosive can be used for good work. Because in recent past, we are hearing 2 days what explosive can do. So this is the area, because there was one of the tasks was there. Ulve Hill is also flowing in that area. It was one of the tasks that we should divert the Ulve Hill, because they wanted to have 3 runways, and each runway have more than 3 kilometers of length. So one of the major tasks was to divert the Ulve Hill also. The proposed airport, the detail is given here. It is 35 kilometers from the existing Chhatrapati Shivaji terminal, and it will be able to cater around 50 to 60 million passengers per annum. And they also wanted to have a very big park of around 610 acres in the nearby area. And the river has been diverted to flow just behind the airport. So nicely, because on the inauguration, they invited us also. I was also there when the Honorable Prime Minister was there. It has nicely been developed, but there is a lot of scope for development, because they have acquired now too much land. Around 7,200 acres of land has been acquired, and that will be, once it will be totally completed, it will be one of the best airport in Mumbai, and similarly at the Jewar in Uttar Pradesh. So just to have the output, that there will be 3 terminals, terminal number 1, 2, 3, and this area, just you see overall area, how it will looks like. There will be shopping mall, complex, there will be hotels. So area-wise, it is one of the biggest airport. Now they have. So we started blasting area in package 1, package 2, package 3, and package 4. So first job was there to have the package 1 and 2. It was completed in 2019, then package 3, then package 4. So we completed it by 2023. Just I will show some of the blasting designs. What? So we classified the total area that how we have to move, because we can't control blasting, that is expensive in all the areas. So buffer zone within 50 meters of the residential structures and important ongoing infrastructure of the project, we decided that it will be by non-destruction of explosive, that will be plasma blasting, chemical blasting. Then complete area between 50 to 100 will be the muffle blasting with a small diameter of jackhammer hole, that is 32 mm, and control blasting of 100 to 300 meters of the structures with shock tube initiation system and electronic detonators. And beyond 300 meters, all the blasts were normal blasts with an ammonium nitrate and fuel oils. We are listening in the news since last 2 months. Ammonium nitrate itself is not an explosive, sir. Just I wanted to confirm it. Ammonium nitrate is a fertilizer, but once it is mixed with fuel oil, that is diesel, in the ratio of 94% ammonium nitrate and 6% diesel oil, it becomes explosives. So only ammonium nitrate is not an explosive, but once it is, carbon is given to them for the chemistry people, once you give the carbon to the ammonium nitrate, it becomes explosives, and it can do any disasters, any constructive work, whatever you want to use it, sir. So joint mapping and profiling of blasting area was done. Blast design, control blasting, and normal protection blast was conducted. And 4 of the staff, PhD students and technical students, were permanently posted, and scientists used to visit for a week in that area. I am happy to say here that 2 of our students, Ramvilas Paswan, Ranjit Paswan, and Himanshu, got PhD based on this experimental work, sir. What will be the safe explosive? So we use that our code from the other researchers, that what will be the optimum amount of energy required to give the armor rock. Because one of the concern was of the people that for the filling of the Ulve River, we should give the armor rock size of 300 to 700 mm. God Sahib is knowing that it was a very difficult task, that each and every blast should produce minimum 60% of the rock size, fragment size, in the range of 300 to 700 meters. So joint mapping, fragmentation size mapping, everything was done, and I am happy to see here that Ulve Hill, total Ulve Hill, was filled with this type of armor rock, and the river was diverted in the peripheral area of the airport. Monitoring of ground vibration, because there is a lot of residential areas. I will show the views. Noise was also monitored for each and every blast, fly rocks, equality assessment, and safety standards. All their methodologies. Okay, I will play from here. The type of rock is the basalt. Volume of the hard rock was 62 million cubic meter. So length of the hill was 2.1 kilometer, and width of the hill was 1.2 kilometer in the central region, which was blasted. Height of the hill to be flattened, it was 84 meters. So we started from 84 meters and came down to the level of the river. The time period given by the authority CIDCO, that is the CIDCO, was 2023. So whatever requirement was there, we completed it. Battery is not there. Okay. So just you see the view of the hill. There were basalt mining was also, stone quarry mining was done in a hazard way. Because when we visited the site, we saw a lot of crushers without any permissions, and mining was still going on. It was difficult task for CIDCO to stop them, but finally they convinced by their resources. And we also monitor the in-hole velocity of detonation of explosives for experimental purpose. We simulated the blast and how much fragment size is required, that is also given in that area, sir. So Ulve Hill, just you see, in both the flank, it is flowing. And now it is the runway of 3,900 meters. So initial view I am showing you, and I will also show what is the final view. This type of cliff, we faced more than 100 cliffs. It was very difficult to blast. It was lying there. But I will show some videos if time permits, and chairman allowed me to give me 2-3 minutes more. Then we also successfully demolished all the cliffs, and now this area is free from any cliffs or any unwanted rock formation. So there are a lot of vibration standards. Our institute also did it, and we have given that what should be the standard, and now we are following our standard, because more than 300 documented data, we constructed the structures in the coal mines, and we studied for 4 years, and accordingly we given the standard which was accepted by regulatory agency in India. So we followed our standard, and accordingly all the blast data were recorded, presented, because every day our staff has to give that what is the recorded data to the authority, because there was a committee was constituted who was monitoring our blast design and outcome. Initially, we optimized blast with design, whatever the data was coming from the softwares, but we experimented it, we modified it, and we also modified that how much time is required for moving of one hole from the other hole. Because most of people are not familiar with it, just you see that it is a geometry blast. So if you are initiating the blast from here, if we are not giving proper time to move from here to here and then here, that there will be ejection of fly rock. And it can go from the top to 500, 600 meters. And that will be disastrous, sir. So it was a very difficult task, sir, how we have to give the distance between the two holes and between the two holes. So that is called burden and spacing. The hole diameter was 150 mm. Number of holes, it was 80 in a particular blast of this, and bench height like this. So all the data whichever had been modeled and based on our experience, the designs were given and implemented. Blast vibration signature was recorded. One of my colleagues, Professor Datta, was saying that in earthquake, 10 hertz natural frequency can become. But we recorded the frequency in the range at 10.1 to maybe 150, 60 data, maybe here, I am not remembering exactly. But in earthquake, more or less frequency is in the range maximum up to 2 hertz. This is the signature what we recorded due to blast vibration, due to dynamic loading. It is only phenomena of 2 to 3 seconds, sir. So what should be the scale distance so that vibration is well controlled? This type of analysis was done by us. So I am not going to in detail. Rock fragmentation size, as I told you, it was done, and 60% of the rocks were of armor rocks in the size of 300 to 600 mm, and that was filled to a level different. Just you see. We also monitor the velocity of detonation of explosive. Just to have the view of audience, this is ammonium nitrate fuel oil, and we have given some emulsionizer. So this becomes emulsion explosives. And that emulsion explosives, what is the rate of burning, how disastrous it is, how much gigapascal energy it is detonating, it is imparting, all were recorded, sir. So this type of wave, more than 4,000, more than 5,000 of velocity of detonation of explosive was deployed there, and accordingly, analysis, I am not going to this part. I will play some video that will be of more important. V1. So the blast which were performed from the top of the hill in initial stage. because it was a very difficult task that there should not be ejection of fly rock, otherwise it will travel height and distance, it will go to the nearby surrounding areas, because people are not leaving the site. We started blasting on the instruction of NI CIDCO. So after 1 year, people were little bit, after getting compensation and everything, they got shifted to other areas. Some view of blast. It is not a control blast, because if you are going with control blast, electronic detonators is costing too much. It was a general normal production blast, but expertise of our scientists and project fellows, they conducted nicely, sir. This is the Ulve Hill, now came down, and near the Ulve Hill, also we blasted it, and that hill was, that's the Ulve River. That Ulve River was filled with the armor rock of this quarry. Only 1 minute, sir. This was a difficult, you see, the traffic is going on. It was only around 60 meters from this area. So all the traffic and this type of cliff were there, and we had to blast it because rock is very hard. So we stopped the traffic, and we did this. Come here. So finally, the land development of Navi Mumbai International Airport was completed safely, following the recommendations of CSIR Simper. It involved 8,550 blasts and 19,665 blast vibration data were recorded and were submitted by signature of our scientists to the CIDCO. And finally, it was inaugurated on 8th October 2025. Thank you very much. Now, this is the view, overall view of the international airport. And after 4 years, when all the parks and everything will come, it will be one of the best airport in Indian history. Thank you very much for your patience, sir.
Thank you very much. So we can have two questions. Yes.
The rock which you got after the blasting, how it was used and removed?
No, there was, because armor rocks generally, we got to the center of area.
Okay.
In the center of area, we did fragment mean size, fragment size, and area-wise analysis. I could not show you.
Because when you are removing the whole hill, a lot of rock is created. So how you dispose of that? Or how it was used in the build-up of the airport?
All the maximum 60% of the rocks were for the Ulve River, for diversion of the Ulve River.
Okay, okay.
60% of the rock was there. And remaining rocks, they sold it to the stone quarries.
Oh, okay.
So when this river is diverted, is there any danger of flooding?
Sir?
Is there any danger of flooding once you do the river diversion?
No, we don't face, because the water in the river was only 5 to 6 meters, sir. It was not very deep. And it was spread around 300 meters in the width side. Lengthwise, it was too much. So we don't face any problem, because we provided the rock, and their civil engineers and everything, they were doing all the things. But we also visited there and saw how they are doing, sir.
I have a short. I've heard that people use lasers, carbon dioxide lasers for making holes in rocks and so on. Carbon dioxide lasers.
Yes, yes. Because whatever the explosive we use, we tested it in our lab, because CSR Simper is having this only one laboratory for testing of explosive quality and their side effects, noxious fumes and everything.
I see.
So it was tested accordingly. We gave the design to the explosive supplier, that is the Solar was the major supplier, and they manufactured it. We got tested it in all this cannon in Simper Dhanbad, and accordingly that explosive are used.
I see. Thank you.
Okay. So thank you very much, Dr. Singh, for a very nice presentation and very informative. Yes. the river has been diverted. So this is, it was a small river, so I think it is not a, it's not Ganga or Jamuna.
Where's the screen here? Oh, here. No. I can use this key now. This one? Okay. So, very good morning. So let me thank Indian Academy of Sciences for giving me this opportunity to do some outreach here. This is not a very regular talk. So what I will try, I will just try to give the glimpses of the activities that we do at National Atmospheric Research Laboratory. But before that, I'll go a little bit to the background that this particular laboratory was started by establishing the 53 megahertz MST radar as national MST radar facility supported by 6 government departments in 1993. And this is an autonomous organization under the Department of Space, and in 2005 it was renamed as NARL. So the radar was, even now it is, the second most powerful radar in the world of this kind. On the right, I just want to show that you can see Professor S.C. Dhawan, Professor Daniel, and Professor Mitra, A.P. Mitra. They were discussing in the early phase of setting up this radar, and that's what I was mentioning on the first day. And in 1990, the first workshop happened, but it was with the partial system that was developed by Shamir, Mumbai, and the first kind of results were coming and they wanted to have the discussion. But actually, NMRF was not actually set up then. So it is only in 1993, it was registered as a society under Department of Space. So if you look at the activity that we have is the basic and applied research on the atmosphere and ionosphere of the Earth and other planets. And we also do develop the technical R&D and instrument development, both ground-based and space-based. We have also a good PhD and postdoctoral program, about 50 students we actually support. And we also do collaboration with academia, education, and training. And since it was a facility, so the charter was to actually facilitate all the scientists across the country and also the scientists from abroad. So that way, so this spirit continues. And one of the important activity of NARL is to provide weather observation and the forecast through modeling for all the rocket launches from Sriharikota. And we also do parameter retrieval, satellite data retrieval, and also validation of satellite data, that is the ground truth validation. Now I'll just quickly run through the kind of activity without going into the details. So the atmospheric boundary layer plays a very, very important role for all that we are talking about, the weather and climate, and also many other ecological issues. So this is something that we do. We also work on the aerosol and trace gases, which are an important component for the climate. And clouds and precipitations are the most important subject of, for example, the cloud microphysics or precipitation microphysics that we deal with, and we have a diverse climatic zone in the country. We do weather and climate modeling, and we have developed in our own center 445 meter resolution, that is a very high resolution weather model, which are very, very important for doing high resolution forecasts, especially important for Sriharikota launch, where you want to tell about whether lightning is going to happen, what kind of wind and wind shear going to load the rocket. So we have, this is the actually prime objective with which we started the activity, that is the atmospheric structure and dynamics. Soon after the ozone was discovered and a lot of curiosity came. And then people wanted to know a little bit more about the middle atmospheric processes which control many, many things. And ionosphere and space physics, that is another topic which we do. So we study the Earth ionosphere and the way the Earth ionosphere is affected by the solar activity called the space weather. And this is so important today that many our communication navigation systems are affected by the ionospheric weather. So, and then of course, part of the space physics. And this is the new element that we have introduced recently, the planetary atmospheric and ionospheric research. And as you know that ISRO is going ahead with big plans for exploring the other planets, including Moon. So this is another area that we want to pursue to understand the atmosphere and ionosphere of other planets, and particularly Mars and Venus, that we have been doing. Now, this is on the science front, and then we also do develop in the campus all kind of radars, and you can see quite a bit of the radar activities that we do, right from 53 megahertz, 30 megahertz. On the other side, you have 1.3 gigahertz, the wind profiler. Recently, we have developed a 445 megahertz wind profiler, and this is the L band, dual polarization Doppler weather radar, and this is the first of its kind in the country. We also do a lot of LiDAR development. You can see all small to big LiDARs to measure temperature, humidity, not humidity. For example, the metallic ions, and then wind, ozone, and aerosols and clouds. We also do the lighting-related research with the climate change, how this lighting activity can actually, bring us some important information about the climate change research. And this is the petascale, 1.7 petascale computing machine which we are using for weather forecast, atmospheric modeling. And this is the data center that hosts all the data that we generate through the instruments, and also the data that we generate through various modeling. So NARL today is not confined to the place which is located 40 kilometers from here, Gadanki, on the way to Bangalore, but we have actually set up many observatories in the country, as you can see, from north to south, from Srinagar to Trichy, and on the east also we have in Kolkata. So this is the kind of length and breadth we have, and we have all radio, optical, in situ measurements, including balloon, tethered balloon. So this kind of experimental facility we have developed. More than 50 instruments we run from Gadanki with plus a high performance computing machine. So this is another program that we have developed after the MST radar became successful at Gadanki. These 5 radars have come up in the country. So this is one is Nainital, Guwahati, Haringhata, Gadanki, of course we have, and then Cochin. And I think more than 70 radars are going to be built in the country for the Indian Air Force. And then, of course, there are about 25 radars are going to be built by IMD. So there are a lot of activity in the phase data system in the country. So a lot of trained people are required to operate, maintain, calibrate, and interpret data. So I little bit give some idea about that, what this radar is. This is a 1024 Yagi antenna arranged in a 32 by 32 matrix. And each antenna is fed by each single transmitters, individual. So we have 1024 transmitter, and the total power is more than a megawatt now. When we started, it was 2.5 megawatt. But these are all solid state. We have already changed from the high power tube version to the solid state. And it can measure actually wind up to about 25 kilometers. You can see here, you can put the beam in the east, west, north, south direction. And simultaneously, you can also fire multiple beams to actually study wind fields in a very short time. For example, in every 10 seconds, you can measure the horizontal and vertical winds, wherein you can study the finer scale processes like turbulence or gust or something like that. You can also get the precipitation echoes, which you can see on the right side, the clear air and precipitation echoes, which come from the Bragg scattering or the Rayleigh scattering. So you can study a lot of things. So here, this is what is, we study the cloud and precipitation using this radar because you have distinct echo and you can characterize them. You can also see the melting layer which is there, and then what it is composed of, whether it is only water or it is ice. And of course, when you have the polarization measurement, you can also study whether it is ice or from the depolarization ratio. So something like that. Of late, we started actually investigating that how this warm rain happens in our tropical system. And the question that has not been resolved is that when you go up to like 10 micrometer, I think everything happens in the normal process, but from 10 micrometer to 50 micrometer, there is no simple pathway. So we are trying to understand whether the neutral turbulence plays an important role in actually forming from 10 micrometer to 50 micrometer. After that, things will go in the gravitational process, so you can have the rain process which will happen. So this is something which is very important for the rain to happen. It is not that the cloud is there, so you are going to have rain. So you see the beautiful, the Kelvin-Helmholtz stability that is coming in the troposphere, which is basically due to the shear flow in the background wind, and beautifully reproduced here in the ionosphere. You see this is the actually cloud picture at a very low altitude, about 300 meters by the FMCW radar, and the same picture actually you can see in the ionosphere, which is done by the KH process. I'll quickly run through. This is actually a discovery, which is the photoelectron-induced upper hybrid plasma waves, which is responsible for the ionospheric echo. They have tremendous potential to measure the electric field of the ionosphere, and we have brought out that how it is inversely related to the solar UV flux, and in a way that how this UV flux actually is controlling the dynamics of the echoing process, including the gravity waves and the tides. So one can also measure, one can also study about the moon echoes, and also not only the moon-bound echoes. Actually, what you see here actually the Chandrayaan-3, and that was detected by the radar. And actually, you can see how this Chandrayaan is actually accelerating and the processes which is going through. So it has a tremendous potential. So this is another radar which is 30 megahertz radar, and actually it can span up to plus minus 50 degrees, a big sky, and you can see actually what is happening in the ionosphere from a space perspective. And we have come up with actually a prediction capability which goes up to 99, pretty close to 100% in a deterministic sense, not in a statistical sense. That is something remarkable. These are the kind of lidars we have. We have temperature measurement, we can measure sodium, and this is actually measures wind, which you can use to study the dynamics, particularly the wave dynamics. And this is the new thing that we have developed, the differential absorption lidar, wherein we have two wavelengths, 289 and 299. So the differential absorption, you can relate to the measurement of ozone. And this is the one which has been developed. You can see how the cloud patches are actually moving. You can study their polarization, so you will know about the water versus ice. And this weather forecast we are doing actually is a test bed in Chittoor, because we have all the facility there. So we have developed this model, 445 meter model, and then trying to develop some kind of what you can say the validation, so that this model actually can be given to the IMD for their purpose. That's the objective with which we are doing. And so this is the kind of modeling that we are using here. This is something new thing that we have developed. If you have 4 to 5 GPS receiver separated closely, you can actually measure water vapor continuously in time. So it's an all-weather water vapor profiler. So if you do that, so your weather forecast is going to be much better, because water vapor is the one which is heterogeneous and plays a very important role in distributing the precipitation system. So these all AIML applications, we are able to derive we are able to predict the electron density in the ionosphere. We are able to do water vapor and also the storm using the GNSS system. This is a new observatory that we have set up in Hyderabad to understand the impact of rapid urbanization and to assess that how this actually urbanization is going to modify and other things, and how the cities have to be developed in that sense to make the cities livable. And this is the Kolkata Observatory. So this is actually in a convective zone to transport material up. So how this material is transported up and then changing the climate is something that we want to study. This is the same thing actually. This is being done from TAFR balloon facility, and also we are running from our place. This is the new thing that the CORS network by the Survey of India, more than 1,000 stations. And we are able to get the total electron content and water vapor, which is going to actually use for the space weather modeling as well as the modeling of the weather. So this is the future program. So this is called VERTIS. This is going for the future mission. This is going to be the ground-based system and for the cloud-based study in the Western Himalaya. And we are going to set up atmospheric climate observed in Brahmaputra Valley to see how these contents are actually conserved in that region. So I think with this, thank you very much. Sorry for the overshooting of time.
Thank you so much, Dr. Patra. We have time for just one quick question, please. We are running a little bit. So.
See, before this institute came into being, how was Mausam being, predictions being done?
That is all done by the IMD. So we are actually not into that game for the country. IMD continues to do that. But the prediction that you have seen, the improvement that you have seen by IMD, it is coming basically from the improvement in the research, which is done by IITM or many other institutions. So we want to contribute through research for the improvement of that, but we do not want to get into that business.
Okay, so but then next I just want to go. How precise is our predictions compared to, let us say, British? Because there, I mean, the Mausam changes so quickly, but they predict it so, so precisely. You cannot. I mean, how is it, with all this?
I would say that in the recent past, the prediction has improved significantly. I think it is going to improve with the kind of thing that I am telling. The number of experiments they are going to put up in different ground-based experiments, satellite-based experiments.
And last one, this PhD program that you are running, are you a deemed university status?
No.
Then
actually, most of our students, they are actually registered with IIST Trivandrum, and some of them are in Andhra University, some of them are in SV universities, and different Pune University, things like that. So we are open to go. We have MOU with them, so they are registered there. But most of them are in IIST.
At that note, thank you so much, Dr. Patra. And of course, you're free to discuss over lunch. And so may I now request Professor Parthasarathy to chair the next session, please.
So, good afternoon, everyone, and I'm pleased to introduce Professor Ravi S. Nanjundaya. We call him Professor Ravi. And just very brief, so Professor Ravi, currently professor at Center for Atmospheric and Oceanic Science and Divecha Center for Climate Change. He was earlier my boss. He was, his director at IITM. And, since 1990, he is doing actually global climate modeling, the seeds he has sown. We have developed a 6-kilometer global model now, but we've kind of followed his footsteps. He was not having any HPC that time. We have the privilege of having HPC now. Welcome, Professor Ravi, and very interesting talk. I heard it in Ashoka, I think. Heat stress, similar one. Heat stress forecast, we all will be interested. Floor is yours. I think, I can take. Please, go ahead.
This one is Good afternoon. Thank you, Dr. Partha, for the nice introduction. I should say that Dr. Partha himself is one of the major architects of a global forecast system, which is now running at 6 kilometers, what's called as the Bharat forecast system. And he's a major architect of it, and it's one of the highest resolution global forecast systems in the world. And I think its quality is comparable to the rest of the world. And to answer the question which I think somebody asked, I think the forecasts in India are much better, and why it is we think the Europeans are doing better, I think we can discuss it over lunch. Okay? Okay. So coming back to the work that we're talking here, it's about heat stress forecasts and its importance in presently changing climate. We know that climate is changing. So let's see how we can kind of use different techniques to improve the heat stress forecast. And this is basically a collaborative response that we are doing, where we are trying to bring in together climate scientists, health experts, doctors, disaster managers, policymakers, engineers, and frontline workers. And this is an initiative being led by IISc and the MS Swaminathan Foundation. And this is the whole set of people, which includes fairly well-known people like Dr. Soumya Swaminathan, former DG of WHO. And of course, yesterday, I think Professor Vijay Chandru had already given a blurb about my talk, and I hope I'll live up to the expectations of this one. So without further ado, I'll kind of go to what we are going to discuss here. I'll give a brief overview of changing climate, then how it will affect heat stress, and how we can leverage AI for heat stress forecasts. So climate change, the very basics. You can see how the temperature has changed over, let's say, I guess, 60 or 65 million years. And what I would like to say is that if we were, the climate change at the end of this century would be what would be about 50 million years back. So in the blink of an eye, we are turning the climatic clock by about 50 million years. That's the kind of change that we are likely to get. And if you're looking at an interim period, the temperatures are more or less constant. And if you look at the more recent period in the common era, it's quite constant. Only after about 1850 or 1900 did it start increasing. And today, if we were to look at, we have increased about 1.5 to 1.6 centigrade as compared to the 1850 values. So this is another picture, kind of a enlarged picture of the changes in temperature from different sources. So if you were to look at climate change, why is temperature important? It's not the only thing that we need to look at, but it kind of gives you a kind of a feel for how the climate is changing. It gives you a direct measure of the trapped heat, and it has a robust and long-term record, which you can easily look at. You can get it from various sources, how the temperatures are changing over the last million, or let's say the 65 million year is basically from a reconstruction. So you have a fairly robust and long-term record. And when we talk of policy, we can easily say that we are going to kind of, right now we are talking of a 1.5 degree containment. I don't think that's going to happen. We are already overshooting the 1.5. Last year, I think it was already at 1.6. I hope we'll be able to contain it to 2 degrees. Okay, and it's also a fundamental driver for other changes, such as melting of ice, sea level rise, evaporation, atmospheric moisture, etc. I won't go into the details of that. So if we look at climate change, of course, there is natural variability. You have climate change, which is human-induced, and also quite often we talk of local climate change and global climate change. So if you're looking at a feature which is worldwide, that's going to be the global climate change. If you're looking at a specific region or a city, that's going to be the local climate change, and the drivers for them are likely to be different. Global scale activities like changes in greenhouse gases, etc. And of course, land use, land cover, etc. That could be the local climate change effect. And quite often, when we talk of climate change, when we are looking at mega cities like Bangalore, I think the land use, land cover change which we are seeing could be kind of more than the global climate change which could be happening. But both of them are we cannot separate out. It's nature, so you cannot separate out the two. But quite often, when we look at regional cities, let's say in city, and say that, okay, it's warming, it's partly the many factors that come into picture, which could be the urban heat island, etc. And I will not go into the details of it. But if you're looking at the impacts, global climate change is going to be widespread, and it has a long-term trends, while the local climate change is varied, location specifics, etc. And these are some examples. We talk of urban heat in Delhi or wildfires in California. And there are many other factors which are related to local. And without going into the details of it, we'll move on to what's of interest to us, which is the heat stress. These are some other basics. And quite a few people say that the climate change that we are seeing could be related to natural variability. This particular picture will tell you that if you just look at the natural drivers of climate variability, you will not get the kind of picture of increase in temperature that we are seeing today. That kind of very categorically tells you that it's human-induced climate change, which is what the warming that we are seeing today. And this is details of radiative forcing. We think greenhouse effect, it's a word which is kind of a bad word in today's world, but it's also a good thing for us. If there was no greenhouse effect, we would be at a frigid -18 Celsius. Today we have it at a more comfortable temperature of about 15 Celsius. And the kind of things that we are going to do is going to be over and above the 15 degrees that we get in the long-term equilibrium, and that's what we are calling as climate change. This is a brief overview of how we study climate and its variability. One of the major things that we use is the Earth system models, which combines ocean, atmosphere, sea, cryosphere, even land, various effects of land. They're all put together into a kind of a co-evolving set of models, which we call as the Earth system models. They are perhaps the most comprehensive and sophisticated models to study climate. And these are the major tools, or this is the only crystal ball that we can peer into and say that, okay, this is how the future climate is likely to be. And it's also one of the major tools that's been used by IPCC, the International Panel for Climate Change, Intergovernmental Panel for Climate Change, sorry. And its most significant statement, it is that it's unequivocal that human influence has warmed the atmosphere, ocean, and land, and widespread and rapid changes in the atmosphere, ocean, and cryosphere and biosphere have occurred. In simple words, it means that we are already experiencing climate change, and that's due to human activities. And these are the other details, which what I would like to mention is that we are close to breaching the 1.5 centigrade level. Last year, year-to-year basis, I think we were closer at about 1.65, but usually when we talk of any particular level, it's a 5-year average. We have not reached the 1.5, the 5-year average, but I think we are more or less going to breach it by the time we reach 2030. And this is how the scenarios of global temperature into the future looks like. If you go with business as usual scenario, you will end up with a 5-degree warming. If you go with more conservative, or let's say more sustainable ones, we may be able to limit it to about 2 to 3 degrees Celsius increase. So we have to be very, I think, aggressive, or let's say I guess now the COP30 is kind of convening, and I hope they come up with a consensus to kind of keep the global warming, the temperatures, more into reasonable limits. And this is what and if you want to see whether the ESMs are giving you a realistic picture, this is the simulated warming at 2100 with 1 degree global warming, and sorry, for 2 conditions. This is what the observation shows, so it's largely in good agreement. And one of the things that you see that the warming is less kind of, you could say high, less you are seeing over in the Indian region, and that's because of the compensation of aerosols and GHGs, which might have reduced, which could have kind of reduced the heating. But in future, if we clean up our effects, and let's say if we want to say that, okay, breathe better, then we have to reduce the aerosols, and then we may see much more global warming, also affecting India also. And this is how the pictures will look like when at 1.5 simulation, 2 degrees and 4 degrees. Basically, you see that the world will be going to be very inhospitable if we were to reach 4 degrees Celsius warming by the end of this century. Now I'll come to the major problem that which I'm going to discuss, and that's heat stress. And heat stress is a global problem. It's not just that it's unique to India, but since we have one of the highest population densities, it kind of stands out. India is one of the regions where heat stress is going to be having a maximum impact. And 2024 saw about 40,000 recorded heat strokes in India. And intensity and frequency and duration of heat waves is expected to increase worldwide. That's what the IPCC says. And of course, if you forecast It helps the societies to be better equipped to understand, prepare, and respond to the evolving changes of heat stress on health. This is what WMO says. And if you were to look at the current forecasting in India, it kind of relies on fairly limited data, conventional models, and on meteorological variables. And hence, we believe there is an opportunity to leverage AI capabilities to improve forecasts, integrate additional weather data, and deepen insights for heat impact on health across communities. That's what we propose to do in this particular project. And if you were to look at how the climate impact on heat-related issues over India, it could face a dramatic increase in the frequency and intensity and duration of heat-related events, largely related by climate change, but also because of local land use and land cover changes, increased frequency and duration of heat-related events, and it's going to be widespread impact. Historically, regions which didn't have experience heat waves or heat stress, such as southern and coastal states, example Kerala and other east coast, they are projected to experience heat waves more regularly. And warmer nights are likely to happen, and higher humidity, which is an even more concerning trend, because that would mean that the nights would be warmer, so you'll be more uncomfortable, and the heat that you kind of gain during the day, you will not be able to lose it very much at night. And there's going to be a major health crisis that could happen. The increased heat could lead to an increase in heat-related illnesses, such as heat stroke, dehydration of chronic conditions, etc. Vulnerable populations could be outdoor laborers, the elderly, and those with pre-existing health conditions. And now that we know that climate change is already upon us, we need to take steps to kind of minimize its impact. We cannot undo it, but we need to kind of mitigate and kind of help out people. So that's what we would like to do, is to create accurate forecasts in short up to 5 days, and up to medium range, that is what we call as medium range in weather forecasting, that's up to 15 days. And it's very imperative that we do this. And this is one instance of heat wave which happened in 2015, and you could see large parts of India were affected, starting from Odisha right up to Rajasthan and up to the border of Pakistan. And temperatures were higher than about 43 to 49 degrees Celsius. And so when we talk of heat waves, which were once rare, now they are now a defining feature of our warming world. So that's what we need to worry about. And if we can kind of improve the forecast that we can do for this, it would be a great thing. And if you were to look at the mortality from extreme weather events in India, what I would like to point out is the last column. And when we talk of forecasts, we've done a fantastic job about improving forecasts for tropical cyclones. There has been a 94% decrease in mortality rate. If you were to remember, those of you who have a long enough memory, or those who are kind of young enough, around 97, 98, we had a super cyclone in Odisha, which I think close to about 10,000 people died. And if we were to look at a cyclone in today's context, I think for the same intensity, I guess the deaths are about 4 or 5, or at best, let's say 10. That's the kind of change that has happened, and that's largely because of improvement in the forecasting that's happened in the last 20 odd years. But if you were to look at the mortality related to heat wave events, or the extreme heat stress events, there has been a 62% increase. That we have not been able to handle as yet. And if you were to look at the number of heat wave days annually at different states, and these are for various periods, and I would like you to see the last column, which is the latest one from 2010 to 2011, you could see that most of the states are experiencing more heat wave days annually. So that is a major problem that we are looking at. And these are some characteristics of heat waves in India. And if you were to look at when the heat waves occur, they usually occur between March to June, with high frequency over north and northwest India and central and eastern regions of India. And if you were to look at the reasons why heat waves occur. There are two types of them. One is the first one is where you have a heat wave over north central India, and it's related to the blocking over North Atlantic. It's a kind of a regional Terry correction that we are talking of, which causes what happens over North Atlantic causes heat waves conditions over India. And the second type of heat wave is over the coastal region of India. That's more towards the eastern part, and what we call as a Matsuno-Gill response. In a sense, it's kind of a response to what's happening over Pacific, the central equatorial Pacific, which we call as the ENSO or the El Nino Southern Oscillation. The causes could be remote. The impact is going to be local. Even if something happens in Atlantic or Pacific, heat stress is going to happen in India. And so we need to worry about how we can do a better forecast for heat stress. And if you were to kind of quantify what we mean by heat waves, it's the heat wave is simply a period of about unusually hot weather, typically of 2 or more days. Quantitatively, a heat wave can be defined as a prolonged period of excessive hot weather, which happens during the summer months. And the temperature increases suddenly as compared to the historical average. And as we discussed, there are many drivers of the heat waves, which could be related to land atmosphere exchanges, feedbacks on soil moisture energy, external forcings, and of course, in today's world, anthropogenic emission of aerosols. Aerosols can kind of cause localized hotter weather, because let's say if you're looking at emissions from transport, the carbonaceous aerosols that come out, they are likely to increase the warming locally. But of course, they have a short period of life period, and they can dissipate out within about 15 days, but they can also cause increase in temperatures. And if you were to look at heat stress and who is going to be affected, the link between death and heat wave is less apparent and more difficult to establish. Of course, the elderly, children, and people with problems, they are going to be particularly affected. And if you are looking at an Indian context, about 75% of the workers, which is about 380 million people, are going to experience heat-related stress. And extreme periods of high temperatures can lead to significant reduction in crop yields. Even agriculture is going to be affected, which could lead to a failure in many crops. And coastal regions of India, east and west coast, are found to be particularly vulnerable to heat stress impacts, which showing a perceptible increase in the number of impact days and a decrease of about 30 to 40% in the work performance, particularly over the eastern region. So it's not just a weather problem, and we have seen that it impacts certainly humans are going to be impacted. And there's going to be a cross-sector challenge which we need to worry about, which is like in the impacts on health systems, livelihoods, and vulnerable population. And there is a data gap. There's a lack of evidence to validate global heat stress metrics for India. This is something major problem. And again, I come back to what we are doing right now, which is a collaborative response, which it has many players in it, which includes government organizations like IMD, IITM, Government of Karnataka, and the State Disaster Management, and of course IISc and other agencies which are coming together to kind of trying to improve the heat stress forecast for Indian region, and initially we are trying to do it for Karnataka, and later on we would like to kind of extend it to the rest of the country. And this is the outcome we propose. So we have right now we are just using only weather data. That's the status as of now. But of course, some details of the person also comes into picture, which we can kind of use. And we use weather data, which is from an AI-based forecast, like particularly of temperature, humidity, wind, and solar insolation, etc. And that the user plus the human heat balance model, we can use that, and it kind of ingests the weather data and gives you risk scores, and then you have a advisory module, which gives you a personalized heat app, watch app, up to about 10 days forecast. The skill, of course, reduces dramatically after you go about 5 days, but it's reasonable in the first 5 days for the forecast. That's what we are seeing. And we are also kind of developing a public-facing dashboard, which gives you a 10-day forecast. Next, what we propose to do is kind of take field data, that is actually go to various sectors of the population, get household weather data and household demographic data, combine it with physiological data, which includes HR, sleep activity, core temperature, hydration, etc., and validate the heat risk score, which would be the first version of it, and then again have a human heat balance model with predicted HR and core temperatures, and then update the generic advisory module to give you a kind of a public-facing dashboard, and then you have a personalized heat watch app, which will take in your information also and give you a more kind of a selective information, which could be more kind of utilizable for the any particular person. So this is kind of a co-innovation and co-implementation that we are looking at. So we are looking at implementation, which is kind of going to be useful for various agencies. The data and engineering is going to come from various sectors, which includes IMD and some publicly available data sets. And the information for climate science is going to come from COS, IMD, and IITM Pune. And it combines data scientists, climate science researchers, and program teams. And the idea is to prioritize and define problems with insights to action, do a data exploration, and design and engineering so that you finally get forecasts which could generate insights. So this is the approach that we are taking. Right now we are starting with a pre-trained AI model and do corrections for that, which includes bias and quantile corrections. I'll come to that in a moment. And then we could either use an individual model or an ensemble model and do a model evaluation, which kind of goes into the parameters for calculating heat risk, which could include temperature, humidity, wind, and radiation. That gives you a heat risk index. And also take data from, in the next step, we would like to take data from secondary sources and create a better targeted heat risk data. And also, we'll also include information from heat vulnerability, heat action plan, and also mitigation strategies. All of them will come together. And what we want to leverage is going to be the AI revolution, which is a quite revolution, you could say that, and which has been trained on decades of reanalysis data. Reanalysis data is like you take all the observations together and put it through a data assimilation system. So you have a cleaned up kind of a set of data which is a proxy for observations, which is derived from the NWP systems. And right now, what we are going to do, the data assimilation, is again kind of linked to the NWP. It's not that we are replacing the NWP systems, but it's kind of using those NWP data, which is coming out of the reanalysis, and learn the statistical and dynamical relationships of the atmosphere so that we are emulating the physical processes to deliver forecasts. The idea is to kind of these forecasts in a matter of a few minutes or seconds as compared to a longer period, which happens with the numerical weather models. And this is the kind of prediction that you see from AI-based model, and its comparison with kind of what we call as reanalysis, which is kind of a pseudo data, the closest you can get to observations. And we find that the 24-hour prediction of a quantity, which we call as the integrated column water vapor, which Dr. Patra also mentioned, which is important, and it has a fairly realistic forecast. This is what the comparison shows. So it kind of gives us confidence that we can kind of leverage these AI-based models to improve the forecasts. And these are the set of forecast models that we are trying to look at. One of them is called the ForecastNet, which uses a Fourier neural operator, gives you an output for 10 days at 6-hourly intervals. And the output variables are temperature, humidity, horizontal wind. There's another one which has come out of China, which uses a vision transformer. It runs for 10 days again and gives you 3-hourly or 6-hourly outputs. And then these are the same variables that are being output. GraphCast is a model which is coming out of Google. And it again gives you a 10-day forecast. Another one is also from China, which is called the Fuxi, which uses a vision transformer, gives you a 15-day forecast, the same set of variables. And the last one with which we are comparing is the more like the gold standard for forecast, which comes out from the European Center. As far as global forecasts are concerned, the Europeans are the best in the business. And it's a gold standard. If you want to compare and see how well your model is going to do, the first question everybody asks you is how well it's doing as compared to the European Center or the ECMWF forecast. So that's what we are going to compare. And these are the results which it's showing. So this is the performance. We are kind of restricting over India. There are papers which talk about, for each of these models, they have given the global metrics. But what we are looking at is for the Indian region. And this is for the IFS high resolution forecast, which is coming at 10 kilometers. Most of the AI-based models are coming out at 25 kilometers. But their skill, even at a coarser resolution, is comparable or superior to that of the IFS HRES forecast. And this is the ensemble mean. Europeans do not do a single forecast. They do a set of 50 forecasts, and then they do an ensemble of that. And this is the one which shows the ensemble mean for weather. And so we can say that we can effectively utilize AI models. And the punchline I would like to say is that if I were to run this HRES model, or the kind of models which Dr. Partha has worked very hard to develop, I need a huge system. But if I take these trained models, I'm not talking of the training of the model, that needs a huge amount of effort. But if I take the trained models, I could just run it on a GPU-powered workstation or a laptop in a matter of minutes. What would take a huge resource to do a forecast, I could just do it on a GPU-powered workstation or a laptop. So that's the kind of a thing that we are talking of. And that's what I mean by the democratization of forecast. Just about everybody can do a forecast comparable to what's like the gold standard. And that you could leverage. And you could do 5 such models together while you are doing only one model when doing here for a numerical model. I'm not condemning numerical weather prediction models. I have worked all my life on NWP models. They have their own good things. But if you were to look at a particular forecast or something that you want to look at, then AI models could be a good alternative. That's what I'm trying to tell you. And if you were to look at which model does better, whenever I'm looking at a 15-day forecast, this is a comparison with what I called as the gold standard. This is the ensemble mean. And the color code shows that which is doing better and which is going to do worse as far as the RMSE is concerned. And on the first day, everybody is doing fine. When you come to the third day of the forecast, you can see that the AI-based models are certainly doing better as compared to the conventional models. And when you go to a longer period, let's say 10 days, Fuxi model, which is the AI-based model from China, seems to be doing much better than even the physically based models. So, and of course, I'm just talking about India and things. I'm not, as I like to emphasize that I'm not condemning NWP models. They have their own advantages, and also, if you were to see, all of them are based on initial conditions which are coming out of a NWP model. So it's till we kind of reach a stage where we can kind of create the initial conditions based on an AI-based system, we are still going to be dependent on AI, NWP-based systems. Till then, I think NWP is going to be useful. And there are many other additional inputs that you get from an NWP model, both for research as well as for improving the forecast, which a conventional AI model may not be able to give you. Okay. So this is the correcting. All models have biases, and the bias is kind of, this is before, just the raw forecast comes from various models. This is from Pangu, this is from GraphCast. And we did what's called as a quantile correction, and the target was the European reanalysis data. And this is for entire India, that's what we have looked at. And after bias correction, the biases are fairly small once we have done it. So which kind of gives us confidence that we can use these models even after doing bias correction, that it's a good alternative as a weather forecast system. And we propose to do this with the IMD data and on a more local scale for Karnataka, where we're using the gridded data from the Karnataka State Disaster Management Center. And this is a typical, just a view of a particular forecast that we did some time back, and we are continuously doing it. And again, coming back, this is the overall picture. So what we have done right now is kind of gone into this particular level. And next, what is I'll talk about is the heat risk index. And the major problem that we have is that these heat risk metrics that we talk of, they are based on all data developed for, let's say, for European countries or America. Like one of the major metrics for heat stress, how a person feels heat stress, has been developed in the 70s for US Army troops. Obviously, we are not US Army troops running around with the same stamina, but those are the heat risk indices that we are using, and that's what is widely used all over the world, which is why we propose to kind of do this kind of a work where we look at various strata of the society and create heat risk index for various people. Let's say the heat risk index could be very different when you are looking at an outdoor laborer as compared to a person sitting in an AC room. So obviously, we need to have a proper heat risk index. That's what we need to look at, and that would also come to the mitigation strategies that we are talking about here. And one of the things that we are also doing is going to be a targeted heat risk dashboard, which I'll come to that in a moment. And the major problem that we are seeing is converting these forecasts into heat risk stress information. We can give you good forecasts of meteorological data, but converting it to heat stress, just as I said, is a thing that needs to be understood. And the conversion needs estimating heat stress, related discomfort, assume the physiology of a person, including their age, nature and of work and disabilities. As I just said, the indoor worker may tolerate heat stress much better than an outdoor worker. And also tolerance could be very different for people of different ages and coming from different parts. For example, if let's say European comes to India and he finds 35 degrees Celsius very uncomfortable, and it's going to be heat risk for him, but we can happily walk around and be very comfortable. So it again depends on where you are coming from and what is the what acclimatization that you have done. So these are various things that need to be taken into account when we talk of a personalized heat risk forecast or an assessment. And as I said, the current conversion formula are based on those conducted on US troops way back in the 60s. And so we need better estimators for Indian conditions that need to be developed, and we propose to develop this information. We are trying to use a two-pronged strategy. One is to improve forecast of the meteorological parameters and also improve the conversion of these into heat stress parameters. And these are some of the heat stress indices which are commonly used. One is called this is called the IMD's heat wave alerts, which just uses maximum temperature. And it's simple and useful for broad alerts, but it has limited use because it misses key drivers such as physiological stresses and even a major driver such as the humidity. For example, if you're in a coastal city, let's say like in Chennai, the temperature may not be very high, but the humidity is going to be very high, and hence you are going to experience higher heat stress as compared to a dry period with the same temperature. Let's say you're looking at Jaipur with 35 Celsius as compared to 35 Celsius in Chennai. You're going to be a lot more uncomfortable there at Chennai as compared to Jaipur. So that's where humidity starts playing a very major role, and that just a simple alert based on temperature is not good enough. Then there is an excess heat factor, whose major components are present and acclimatized temperature, and it identifies hot spells for early public health warning. It has its own uses. Then you have the heat index, which is an operational warning in USA, which includes both temperature and humidity, and measures perceived heat in shaded conditions and underestimates stresses in direct sun or wind. And there is one more which is very commonly used, which is called as the wet bulb globe temperature, WBGT. And it's an operational warning which is being used in Japan. It includes temperature, humidity, wind, and solar radiation, and is widely considered to be the gold standard for assessing physiological heat stress, which is widely used in occupational safety. And then you have the Universal Thermal Climate Index, UTCI, whose key component is a physiological model, best suited for evaluating outdoor thermal comfort and public health risk. And these are the formulae for various the various metrics that just now I discussed. And as I just said, existing heat stress thresholds in India mainly come from studies in developed countries and may not reflect Indian conditions. So that's something we need to worry about. And that's what we are trying to develop with, in collaboration with the Swaminathan Foundation and other institutions to kind of do these field observations so that we get better India-specific estimates of the, which is relevant for our country. So now I'll come to the forecasting for heat stress in Karnataka, which we are using as a test case. And it's actually a very good test bed if you were to look at. Karnataka has a very heterogeneous region. It has coastal Karnataka, which is very humid, and southern Karnataka and northern Karnataka, parts of Bidar, etc., which is hot and dry, and of course southern Karnataka, which is very comfortable. So it kind of has various climatic zones which are kind of useful to kind of developing heat stress indices or do heat stress forecasts. And once we use it as a test bed, we can go to kind of replicate it in other parts of the country. And these are the heat stress parameters which we just discussed. And this is over a dry interior region, and these are the various indices you would like. And since you are looking at a dry region, the WBHD is not very, these are for two different years that we have calculated it. And you can see that year to year there is a variation, but this is just an estimate that we are taking from observation. It's not just of a forecast. For a moist coastal region. And this is I would like to, can I click on it, please? I want to just show a quick, what we are looking at as a heat health dashboard. Yeah. This is a real-time heat forecasting platform, and you can look at the current forecast. And you can choose any city if you like. Let's say you take, okay, I can't see, it should have Bangalore somewhere, but let's say we take a hot place like Bijapur, and it tells you where the heat stress is likely to be forecast, is likely to be more high. And let's say you look at a place like Bidar, where the temperatures are higher, and this is a 10-day forecast, which tells you the heat risk. Of course, right now you are looking at a fairly comfortable season, so there's not much of a heat risk, but there's some kind of a heat risk over northern parts of Karnataka that you could see that. And the data from that, you could either generate a report, and you could also download the data, and this data is kind of being kind of given real time to the Karnataka State Disaster Management, IMD, and various other organizations. Oh, sorry. How do I come back? Yeah. It should be already there in the. It should already be open, right? It's okay. Just go up. Okay, just I think for Karnataka, what we did is we took, to do a hyperlocal forecast, we kind of combined data from the State Disaster Management Center, created a gridded forecast for that, and combined it, so the spatial, and also combined it with the AI-based forecast, kind of get a better hyperlocal forecast. The AI model gives you at 25 kilometers. Right now, we have kind of taken it to 10 kilometers. Later on, we would like to take it to a finer resolution, where we kind of merge it using a second level of AI. The first level of AI is already there in the AI-based model. Then we would do a second level of AI where you kind of do a hyperlocal forecast, merging the localized data, create another forecast, which gives you better skills. And this is the forecast that you are getting after downscaling of the data. And this is the 2-meter temperature that we are looking at. We have taken data from the Karnataka State Disaster Management Center, combined it with AI-based forecast, and to get a better forecast for the Indian region. And this is the kind of forecast that we are looking at, and various metrics, which includes the scale Tmax, ground temperature, and Tmin. Those are the two major parameters that we look at. And these are some of the metrics for the forecast. This is the raw AI forecast. So this is the error. And after doing the downscaling, that is kind of including localized information and kind of doing a higher, finer resolution forecast, you can see that the errors are kind of reduced significantly from 3.68 RMSE to about 1.86 RMSE. And this is the kind of thing which Vijay also talked of. So basically, you can do a kind of a personalized heat app, which we can use it, and create your own files where you give your other details and also the health kind of parameters, and you can get a 7-day forecast based on this, which will personalize your forecast. Right now, we are just using the meteorological parameters. We are not doing it. Later on, when we actually kind of do the personalized based on the kind of work that you are doing, etc., that will be the second layer of heat stress forecast that we are planning to do in that. So this is the summary. And in effect, I would like to say that we can use AI, and anybody can do AI. It's like a democratization of the forecast that I'm talking about. We're also developing a dashboard, which I just talked about. And we propose, right now we've done it for Karnataka, we would like to do it for the rest of the country. And of course, we would like to increase the accuracy of the forecast. They're not perfect, but we hope to increase the accuracy of the forecast. And all the forecast outputs and data sets, the analyzed data sets, which we have built it ourselves, that's open to public. The actual, let's say, station data sets, because there are some constraints either from IMD or Karnataka State Disaster Management Committee, that we are not making it publicly available, but the rest of the data is going to be available publicly. And we are also improving the spatial and temporal resolution of the model, as I just said. So also having an interactive dashboard, example of which I showed, to disseminate forecast in a simple, actionable format for policymakers and communities. Thank you.
Take some questions? Okay. Yeah.
Thank you so much. I was just wondering, in terms of urban heat stress compared to rural heat stress and urban heat islands, because even within urban areas you can have heat islands, and what your recommendations could be for architects and developers in terms of building design for different parts of the country. We all know that we are aping Scandinavian designs with glass windows everywhere because of, so surely with the level of buy-in you have with the government, could you not take it to policy to inform, building codes, architectural designs, because you are already, at a higher level with maybe influencing these issues.
Well,
okay, I'll stop.
I mean, forecasting is an easier problem than the policy. For example, we know what causes problems in Delhi, the air quality, but there are no easy solutions. So similarly for this, certainly we could talk to the relevant policy authorities, but whether they implement it or not is a question which I don't know, I cannot answer. But certainly we can improve the forecast and kind of create better assessments for personalized heat health. But when it comes to buildings or urban heat islands, of course we could kind of do hyperlocal forecasts. We could do that kind of a work, but giving recommendations and them being followed, you know what's happening in Bangalore. So, I mean, there's violation of laws everywhere, but I don't want to get into it. Okay, yeah.
So I have a question. You may have said this and I may have missed this. What is your definition of heat risk? How do we define risk?
Okay, there are various metrics that I just now
So
pointed out.
what goes into the definition of risk?
Okay, let's say if I were to look at it from a meteorological point of view, essentially it's going to be winds, temperature, humidity, and combination of these. And supposing you want to personalize it, you bring in the person's age, his pre-existing disabilities, etc., and you combine it with some, let's say, some already existing, let's say, the formulae or whatever the research has already been done. You combine that to give you a kind of a heat stress, and based on the temperature and others, you can kind of tell them what is the heat risk they might be feeling. Let's say if you're at 35 degrees or 25 degrees, heat risk might be very low. But if you're going at 35 or 40 degrees Celsius, the heat risk is likely to be higher. But if you're, let's say, young and energetic or athletic, the heat risk could be still low. But let's say somebody like me who's much more aged, the heat risk could be very higher.
No, is that all? Obviously, you're talking about a regression equation.
In
Is
some
all
sense,
of that
those are the
taken
set of,
into account?
Okay, I could, those were the ones which I mentioned, but I kind of rushed through it.
Okay, maybe we can talk. It's getting technical, so maybe we can talk about it after or during lunch break. Thank you.
Yeah, these are some of the equations that we are using for heat stress indices.
Yeah. From your diagram, I think one of the figures was the effect of this one, temperature rise is going to be felt more in the northern hemisphere because that is what was.
Okay. Northern hemisphere is likely is more continental. The southern hemisphere is oceanic. So the effect is certainly going to be more in the northern hemisphere. And of course, if you're looking at the population, most of the population is in the northern hemisphere. So it's going to be having a bigger effect there. And I guess land temperatures would rise faster than the ocean temperatures. So you may, if you're looking at global the variation of temperatures, it could be higher over the northern region as compared to the southern region.
So we are safe. I mean,
we are actually not safe.
No one is safe.
We are in more trouble. Yeah.
Just wanted to make a quick comment, and Rennie, this is related to what you had raised. As part of the team, we also have the Center for Sustainable Technologies, Monto and others from ISC, who are advising on vernacular architectures and ventilation and so forth. And we were also working with CSE and IUDI on actually advising architects on some of these issues. So it's being done as a wholesome project, but yeah.
Sorry, I forgot to mention, we are also working with CSE.
So, like, cyclones are relatively short duration events, right?
Yeah.
So the government can move people out and over the area, but what for heat stress, it happens over probably extended period, and people cannot be really moved out, presumably, in the same way. So how?
That's where I think the advisories come into picture. I mean, if you can give them better assessments and give better advisories, maybe they can take appropriate action to kind of make themselves safer.
But people who are at work, I mean, they probably don't have too much
That's
option.
a major problem. Like an outdoor worker, temperatures are going to increase. It's a major problem for them, but we can only, at this stage, we can only advise, but we can tell them what they can take, maybe drink more water, go into shade more often. Those kind of advisories we could tell them about that.
Two short questions here.
Sure.
Is the air pollution included in your modeling? I mean, the AQI and so on. And number two is why the mortality rate due to lightning went up, 52%.
Well, I mean, okay. first question I can, right now we are not taking AQI into consideration. It's a tougher problem to handle. I mean, many people have asked us about including AQI for heat stress. The two are related. But, you know, just doing plain heat stress has been a problem. So including AQI, which has even more uncertainty when you are going to do a forecast, we have not yet done. At a future stage, we certainly would like to do AQI also. And as far as the lightning is concerned, it could be related to more number of thunderstorms which are happening. And also, like, maybe people are out more into the open and doing farming operations. And in general, the population has increased over a period of time, so the likelihood of getting being struck by lightning could be higher. But I think here is an expert on thunderstorms. You can discuss with him, and he can answer it more.
Lightning issue is we are not able to reach the last mile.
I know, it's also a problem of...
A farmer on the field doesn't carry any mobile. Okay, so whatever way you give the message, it doesn't reach him. And he is the most vulnerable on the field. So that's why you could see 52% rise. That is one of the major bottleneck. We are not, I mean, it's from ministry, from government, how to reach the last mile when a person is on the field. Still it is open question. We could not close.
We are actually, I think IITM is doing a lightning forecast. And they are also kind of putting out on mobile, but I mean, it may not be reaching them.
If you see the slide, you will see the skill has improved, but the death also has improved. These two doesn't go hand in hand because we are, so the skill is not getting translated into the end user.
Same thing is for the people in the sea when they go in the boat.
True. So last question from, a comment from Professor Gould.
I was just wondering that three effects that, considering that risk essentially means, you know, all potential losses that could take place. And I was thinking that three items, and none of these are so easy to quantify. One is the loss of productivity, industrial productivity or whatever. The other is the debility, general debility amongst people. And the third, I was wondering, since respiration increases faster than what photosynthesis could do, what would be the loss in the productivity of the, you know, general? I mean, these will be, I'm sure they need researchers. There are no easy answers to these.
Much research needs to be done. We can only, existing whatever literature we have, we can use it. But as I said, we want to do that kind of a thing where we look at the last mile, look at the actual people for different strata of society and try to get better heat risk, health heat risk indices, kind of tailored for them, let's say for an outdoor worker as compared to people inside, for various, let's say old people, children, etc. We would like to do that. We are going to do a kind of a sample survey in Tamil Nadu and Karnataka, but that is yet to be done. We have not yet started it. So that's the proposed work that we are planning to do.
So thank you, Professor Ravi. Let us give a big hand to Professor Ravi. Send over.
Well, as the As the 91st annual meeting of the Academy draws to a close, this is an opportune moment for all of us to convey our sincere gratitude to all of those who have been a part of this success. For the formal proceedings, I now invite to the stage our distinguished co-host, Director Professor Satyanarayana, IIT Tirupati, Professor Narasimha, Dean Research, Sri Venkateshwara University, Professor Amit Kumar Patra, Director, NARL, and Shri Inderpreet Singh Kohli Paji, Registrar, IISER Tirupati. I also request Professor Raghavan Varadarajan, President of the Indian Academy of Sciences, to kindly come forward to do the honors. May I now invite the Second Secretary of the Academy, Professor B. Gopal, to formally propose the vote of thanks. You can propose the vote of thanks. No, but why they stand on there? We need to, yeah. Okay. As a token of our gratitude, this is a memento to the institution, also with the Heritage Book of the Academy. I request President of the Academy, Professor Raghavan, to first present the plaque and memento to the Director, NARL.
Whichever order it's in is okay.
This is SV University. Our sincere thanks to IIT Tirupati. Our sincere thanks also to NARL. NARL. And our prime host, ISER Tirupati. Please be on the stage for a moment. Okay, sure.
So anyway, I would like to thank all of our hosts, and Professor Satyanarayana has just returned from Germany and at very short notice has come to the venue. So it's very nice to have him here, and thank all of you.
Yeah, so before we come to the close, it's my honor to propose the final vote of thanks. And I've kept this along just in the case I miss some of the names, but even then that I'm prone to making errors, so forgive me. First, of course, Shantanu Bhattacharya, the director of ISER Tirupati, Inderpreet Singh Kohli, the registrar, and the registrar's office, chairs of all the departments of ISER Tirupati, the LOC team, and of course all the people who helped us in getting this in order, and the IT team, and a special mention to Srikanth, who stayed in the Colosseum all along the event for all 3 days. Director NARL, Dr. Amit Kumar Patra, Dr. Satyanarayana, Director of IIT Tirupati, Dr. Tata Narsinga Rao Garu from the VC University for co-sponsoring this event. The media, Innovations India Private Limited, who were a part of the registration team, and they've checked out the Face ID based check-in system, and they also helped us with a couple of coffee sessions. And a big thank you to all, and most importantly, the thank you to the TTD authorities for accommodating bulk darshan at appropriate times. And I think the fellows, all the fellows will appreciate that. And of course, thank you for your enthusiastic participation. See you again in the midterm meeting. Till then, bye-bye. I forgot. I'm supposed to thank my own and extremely grateful to the academic staff who made this possible. And a big vote of thanks to all of them. Thank you so much.